Hello, everyone. Welcome back to Ask a Scientist Gaming, where we combine mediocre gameplay with expert science. I'm Ken Hansen, Associate Professor in the Department of Chemistry and Biochemistry at Florida State University. I'm a photochemist / photophysicist, which is a fancy way for saying I shine light on molecules and they do stuff. Some of that stuff is useful, some of it's not. We try to figure out what that stuff is. But more importantly, joining me today is Dr. Angela Knapp. Angie, if you wouldn't mind introducing yourself.
Sure. Hi, I'm Angie Knapp. I'm an Associate Professor of Chemical Oceanography at Florida State in the Earth, Ocean, and Atmospheric Sciences Department. I study the marine nitrogen cycle, and I use geochemical tools to study biological and physical processes, especially using nitrogen stable isotopes.
And what do you typically teach?
I teach chemical oceanography. I teach, right now I'm teaching geochemical ocean tracers, where we are covering the latest and greatest geochemical ocean tracers to study things like photosynthesis, rates of net primary productivity in the ocean. And then I also teach history of Earth systems. I also teach, I do a ChemOcean seminar, which gets grad students up presenting their research and practice speaking in front of a group.
That's fun.
And I even did a professional skills development class last year, which was something that the graduate students requested on the Diversity, Equity, and Inclusion Committee.
Oh, that's fun. Like soft skills and CVs and interviewing.
Well, it was more than soft skills. They wanted to learn. It was basically a class to get people who weren't comfortable coding skills to learn.
Oh, wow.
MATLAB, R, and Python, which I did not teach.
That's
but
fun.
we brought a bunch of people in, and then the students, we actually opened it up to undergrads and grad students, and then their assignment was to make, they had to identify 2 skills that they wanted to learn by the end of the semester, and then they had to make a video of themselves teaching that skill. And so we developed a skill library, so then those are up online and stuff.
Fun. All right. Equally important. What game are we starting
Super Smash Bros.
for the Nintendo 64? So
Thank you.
anyone hoping for the advanced version?
Old school. Yeah. We are
we don't
all
do
old
that
school
here.
here. Yeah.
Actually, Lance Cooley last week played Overwatch 2 online,
Yeah.
real time. It was interesting.
I don't know that one.
But yeah, playing some Super Smash Bros. And this is the original one, the one that took off the franchise.
Yeah, this is popular in my house, and my 14-year-old can currently beat us all, the rest of us combined versus her. So, and that is, I think, the birthright of teenagers, right? That is their skill set, is video games.
Let's do it. So you're going to do the one-player challenge mode?
Yep. All right. We're going to make it easy.
What's your character of choice? Do you have one constant? Or?
I am always Kirby. And just, for the record, this is a different controller than me. So I am struggling already.
I'll give this disclaimer. Angie's not used to the Nintendo 64 controller, which is what we're actually using here. So
Yeah,
I apologize.
We have a sort of a Raspberry Pi set up in my house, so I can't even. There we go. Whoa. So all the selections are different.
Yep.
So it's going to be good, you guys.
It's going to be great.
It's going to be amazing.
While drinking and talking
Yeah,
science,
I'm so good at all
fine.
these things. It's going to be great. Oh, God.
Probably the A button. I honestly don't know. I didn't play this one that much.
Yeah. If my 14-year-old was here, it would be fine.
Are they going to log in and
No.
drop?
They're doing bedtime. Okay.
Try to do that one.
Hey.
There we go.
That's too easy, please.
Hit that and then probably hit start.
Yeah. All right. That's good, you guys.
Versus Link.
Okay. Oh, yeah, and the 14-year-old likes to be Link, so.
Perfect training. And so, anyone that's a long-time viewer, and this is Angie's second time on the stream. We had to look it up. It's 2 years and 2 weeks ago was the last time she was on.
Yeah.
And it was not peak COVID, but it was like, we were still online classes. We were online everything at that point.
Yeah.
And so the last setup, and this was a battle, is we did it remotely. So what I did is I dropped off a green screen, and we basically had a Zoom setup where the guests would play their game, and we'd transfer it via Discord or Zoom. But we had a lot of issues setting up the...
But we totally pulled it off.
We did, but you were playing on like my computer with a small box in the corner, if I remember correctly.
Yeah.
So like my
I
old
blocked it out. I don't remember.
well, my old laptop didn't have the processing power to do like streaming and
Oh yeah,
at
we
the same
couldn't do
time.
this game, I don't think.
Yeah, exactly. So we did set up Super Mario World and you played that for 3 hours.
Basically.
So, but we're in person now. So we're using my computer. Controller's different, but it's way easier this time around.
Totally fine. Yeah.
Yeah.
Just go with it. Okay. Oh, Yoshi. Oh, fun. Okay. Cool.
Yoshi team. I like it.
That's good. Yeah. Just got to find all my my powers. I was just practicing with my 14-year-old last night, but the different controllers. I got to find out where all my skills are. Oh, hi. There we go. Oh, look at that.
So the
It's gonna be good. It's okay. My strategy is just button mashing in general, so it all works. It doesn't matter what controller, button mashing's the same.
So have you played any of the newer versions of this game? Like this is like professional game playing, like professional leagues of Smash Bros. Players.
No, I don't have time for that.
It's fun to watch though. I mean, if you know a little bit about the controls and whatnot.
Yeah, a little bit. Yeah, that would be helpful. But no.
Yeah, for some reason I missed this game. Like I played a little bit of Bond for the 64, but never played Smash Brothers.
Yeah, I never played this one when I was little. I think so. My husband and 14-year-old have set up like a Raspberry Pi version of all the games, and so I've learned it since we downloaded that. And but this is not something that I grew up playing. So we're saving those for later. When I need more muscle memory.
That's fair. Do you play any of the modern gaming systems, or do your kids?
No, we don't have it. We just have like the Raspberry Pi and the crappy old controllers.
Yeah,
and, like,
but that's thousands of games.
Yeah, no, that would be dangerous. So, yeah. So, no, I think we're better off limiting it.
Star Fox. It's fun.
Yeah.
All right, so you've had a little bit of time to settle in.
Yeah.
So you're just recently back from a trip.
It's true. Yes. So I am leading a 7PI NSF funded project in the Gulf of Mexico. And our overarching theme is trying to understand linkages between submarine groundwater discharge and the nutrients, especially iron and nitrogen and phosphorus that come from it, versus, and then how that relates with phytoplankton growing in the Gulf of Mexico, especially. See, I'm not gonna be able to talk and play games at the same time.
That's
Totally
part of the fun.
Yeah. So we're interested in the harmful algal blooms that form off of Tampa Bay, and we encountered one. We tell lots of stories about that. So we were in the middle of a Coronia brevis bloom, and but also we really, our favorite, and Ken remembered, is trichodesmium. So that's our favorite. So I'm just thrilled that you remember trichodesmium, and we found all the tricho on this cruise. We were so excited.
I mean, when you say in the middle of the bloom, it's just like you show up on the water during a scheduled time and it's just coincidentally thermal cycles are right that...
Well, that's the question. What is causing the blooms? We don't totally know. And so we're both studying that on this NSF project. And then some colleagues and I also have contracts with the Florida Fish and Wildlife Commission, who are tasked with monitoring harmful algal blooms on the West Florida shelf and trying to understand their causes. And so we have a bunch of different cruises, because the thing is the water on the West Florida shelf changes really quickly. And so if you want to try and understand temperature or nutrients or whatever it is that's controlling those blooms, you kind of have to go back. And so they kind of call it sustained monitoring or observations, because you just have to keep checking, because things change so quickly, compared with, say, the open ocean, where historically where I've done a lot of work is out in what we call the big gyres in the middle of the ocean. And there, I feel like you have a better shot at understanding what's going on. But apparently we all wanted to challenge ourselves. So we're working in the shelf ecosystem where things change really quickly. So we're trying to link submarine groundwater discharge as a source of nutrients to the phytoplankton that we see.
I mean, so what's your window on this bloom event? How long is that?
So it varies a lot. And so they usually last months to sometimes almost a whole year. And so they cause respiratory distress for humans. They cause fish kills. They kill manatees and dolphins and all kinds of things down in Tampa Bay. So there's a wide interest in really trying to understand what's driving these.
I mean, so what's presumably the general mechanism is understood. There's a certain concentration of nutrients and temperature, or it's just no one knows right now?
No, we don't get it. And so this is why we're interested. So the thing is that we call the West Florida shelf oligotrophic, which means that the usual forms of nutrients that phytoplankton like to eat, the inorganic forms like nitrate and phosphate, are scarce. They're pretty much at or below detection limits all the time. And so this is one of the reasons that I'm really interested in working there, is because I study organic nutrients and I have tracers for looking at organic nutrient consumption, especially dissolved organic nitrogen. And that's really the only ball game for these phytoplankton out there. And so we're really trying to tie organic nutrient sources to the phytoplankton distributions in space and time.
I mean, so you have an origin point to phytoplankton and then just reproduction, or how does that?
No, we don't have an origin point. No, so that's one of the things that we're trying to figure out. But and then there's physical oceanographers who are trying to understand, the circulation on the shelf and where waters come from and how much they're mixing with different things, but it's super complicated. And so no one knows. Cool. I mean, let us try.
For an outsider looking in, for me, it's a mass balance problem, right? Like there's a nutrient in, and then there's a mass generated.
Right. But here's the thing: we don't have great constraints on those mass balances.
No, that's true.
So submarine groundwater discharge has been proposed as a nutrient source for decades. Oh, okay. Get on it. Come on. And so, I'm bad at this. This is okay. So people have proposed. This is just going to be painful. Submarine groundwater discharge for decades, but there hasn't been a lot of recent work on it, and especially co-locating it with the locations of the phytoplankton blooms at the same time. So one of the things that was really great on this cruise was that our colleagues at the Florida Fish and Wildlife Commission gave us something called an imaging flow cytobot to take with us. And so we call it basically phytoplankton TV. And so it takes on our boat, there's a pipe at the front of the ship at the bow, and it sucks in water all the time. So we call this an underway seawater intake or line, and we plumb that to this imaging flow cytobot, which takes pictures, every couple seconds, and the people who work with that data reference image libraries and then try to map out distributions of phytoplankton in space and time on the cruise. And it was kind of fun. It was in the lab where I was working. And so if we had a minute, we'd just be like, hey, what phytoplankton are here? Let's go check phytoplankton TV. And we saw a ton of Karenia brevis. And when we were in the bloom, so for people who don't speak or think in chlorophyll terms, if you're in a pretty low productivity part of the ocean, typical chlorophyll concentrations are 0.1 micrograms per liter. These ones were 6. So it was like chunky. You could walk on the water. Yeah, exactly. And it was really bad because the crew thought that there was conjunctivitis going around, but basically the scientists were just processing water, and there was so much K. brevis in there that if somebody got one in their eye, they were really irritated, and everybody, especially people who had asthma or were a little bit sensitive, had sore throats, and they were kind of having, irritated, their throats were irritated and stuff like that. But so what we did is we're like, okay, well, we got to get these samples in the bloom right now. This is where our station is. And then I'm like, okay, we're not going to park the boat and spend the night here. Can we get out and go to clean water and spend the night there? And once we did, we felt fine. But yeah, the crew kind of thought there was conjunctivitis for a little bit going around on the boat. But we're like, so, some people.
Man, that's crazy. I mean, what's even more insane is that this presumably happened hundreds of years ago, right? To sailors, and they just have no idea.
Well, this is the question, right? Yeah. And so we don't know. We don't have great metrics in the paleo record it would be great if somebody could find a biomarker for the K. brevis, and I think there are people working on that. But poor Kirby. Pikachu is what my 9-year-old plays, and she loves to like electrocute everybody. She's really good at it. And so so we don't know, and so there are these hypotheses, right? That chuck myself off the side. Florida, there's a lot of septic tanks, and these aquifers sort of, basically I think Florida is Swiss cheese, right? Yeah. And all these aquifers drain out onto the West Florida shelf and in the coastal region. And so the question is, I swear I'm better at this when I have my controller. Excuses. Oh no, Pikachu's gonna destroy me.
Pikachu with a gun.
Exactly. My 9-year-old would be so pleased. Yeah. She'd be like, yep, mom's
canon,
going down. Yeah, exactly. Oh, get the fan. Okay, I have a shot. So we don't know if there were these harmful algal blooms 100 years ago, 200 years ago, or how important, anthropogenic sources are. And so that's one of the things that nitrogen isotopes, we hope, will be able to help us get at. So based on some preliminary work that we've done, we think that there's a distinct nitrogen isotopic signature associated with the dissolved organic nitrogen coming out of submarine groundwater discharge, and that looks different from what's coming in by the loop current from the North Atlantic. And so we're hoping to use nitrogen stable isotopes to track some sources. I also have colleagues who are doing iron isotopes and also characterizing the dissolved organic matter using some both characterized and largely uncharacterized techniques. And so, oh God, Donkey Kong. Okay.
I mean, so if I heard correctly, you mentioned trying to find historical insights. Is that like coring, or how do you?
Would be. That's not part of our work. So we don't have any paleo work proposed. There are people who I think are interested in that, but they're not part of our NSF project or the FWC work. And so I hope they're out there, and I hope they're developing biomarkers for K. brevis. That would be swell. But that's not part of what we're working on. We're just strictly working in the modern. And then we have 2 cross-shelf cruises. So we had one in February, March, and we have one coming up in June and July. And then working with the FWC, they go out all the time. And so there are a bunch of sort of complementary data sets that we're going to have where many of us who are on the NSF project are going to analyze a lot of the same parameters in those sample sets as well.
Makes sense. Calm down, Bronco. Welcome back. I hope grad school is treating you well. Second semester in, almost done with second semester, hopefully done with classes soon, which is awesome. All right, Calm Down Bronco wants to know, if you could research oceanography in Florida, where would you like to go?
Good question. So I have some French colleagues who are very smart. They have field stations in New Caledonia, and they often are going between New Caledonia and Tahiti. And they collect samples for me. And I haven't been on these cruises, and I think this is a poor life choice. And so I really think...
Wait, is that a research-based decision or a personal decision?
I mean, it's both. So I really think I need to be going with them on some cruises. And so actually their work is also about nitrogen fixation and trichodesmium, because trichodesmium forever. And what's really cool is they're seeing, so I sort of feel like I'm never going to get out at this level. But aside from that, my work is sort of moving in. Yeah, it's just pinball here for Kirby.
What's the goal here? It's to
get through this, and then get down. See where those arrows are going down? I'm never getting there, but that's okay. So one of the things that nitrogen fixers need a lot of is iron. And so we know that there's a lot of iron coming out of the submarine groundwater discharge out here in Florida, but my French colleagues are working near the Tonga subduction zone. So I don't know if you guys remember that huge eruption in Tonga, like a little more than a year ago. But there's basically a subduction zone, and there's some shallow hydrothermal vents. that, does anybody know how to get me out of this level? My kids do. I'll take tips. At any rate, there's shallow hydrothermal vents that release a lot of iron that makes it up to the euphotic zone. And so what we're seeing are some of the highest global rates of nitrogen fixation in the area. And so we actually have a paper that's just accepted in Science.
Wow.
So that's going to come out
Cool.
so with the French colleagues are leading. But so some folks who've done work in my lab and I are on there. So that'll be fun. But yeah, so this is pretty cool. So I really think I need to go to.
Can you down B?
Tonga. Can I down B? Down and B at no friends.
Bronco, thank you for trying.
Yeah. I'm not sure if you have access to abilities. No, I don't have access to any abilities, I promise.
In general. Just as a
Button
truth.
mashing is what we're doing here tonight. Sorry, guys. Yeah.
No, that's fun though.
Yeah, I think I need to go with my French colleagues in Caledonia. So, yeah, that's my next goal.
I mean, so in terms of the NSF funding then, do they favor local US related things or?
Interesting. Good question. So, no. And in fact, there is a common perception that NSF chemical oceanography is anti-work in the Gulf. And I don't think that's true based on, obviously, we got this big project funded and we talked with our program manager. Program manager used to be at UT Austin Port Aransas. And so I think she has a particular appreciation for the Gulf. But
Where would that bias come from if it did exist?
I think there has been a lot of local funding, and then there's kind of this old school view that like there's nothing interesting going on in the Gulf. But so then
The Gulf is solved.
which is crazy because there's all kinds of really interesting geochemistry going on in the Gulf.
Yeah.
But and so I can talk more about that. But so I think there's a new generation, like people my generation and younger, feel like there's a bunch of really interesting science questions that they want to ask in the Gulf. And so we're really excited to have the opportunity to try and get at some of this stuff. And there's been a ton of work that's been done. And then a lot of people are just frustrated because some of it's by local or state agencies, but then trying to access those data, some of it's not published, and it's just hard to put all that data together. So there are efforts to sort of link data sets and try and get stuff co-located so that, you're like, oh, what are the nutrient concentrations? I don't know. Nobody's ever talked about it. And it's like, actually what they have, but it's not published necessarily in the literature that we would expect it to be in or wherever. So I think there's a lot of work going on right now to try and, bring a bunch of prior efforts together as well as ongoing new efforts and try and synthesize a bunch of these data sets and figure out what's going on.
So back to the question of Gulf versus the global situation. So I'm naive in the area. So obviously NSF has broader impacts and you have to actually try to solve certain problems. It's not just fundamental research. Like what are the oceanography related problems that they would care about more than say the like algae blooms that affect a lot of people? What's the
So some of the fundamental core goals of chemical oceanography are to understand, like you said, elemental fluxes and elemental budgets. And we just don't even have first order handle on those. And I think, so the reason I love nitrogen fixation so much is because it is the ultimate source of nitrogen to the ocean. And it's biologically controlled, which makes it different than phosphorus and iron, which just come from the weathering of minerals on land. So phosphorus and iron, just like if you go to Home Depot and you get a bag of fertilizer for the plants growing in your yard, it says NPK on it, right? So just like plants growing in your yard need those elements, plants growing in the ocean need those as well. Except they have plenty of, let's replace, lava. Oh no, bombs. Replace potassium with iron. because iron is much more limiting in the ocean than on land, and potassium is limiting in the ocean. But at any rate, plants need all those elements to grow, and phosphorus and iron have strictly physical weathering sources. But nitrogen, the inputs and outputs, come from microbes, which makes it more fun, more interesting, and more challenging, harder, we think. Chemical oceanography is fundamentally interested in trying to constrain fluxes and understand their sensitivities, especially as it relates to the global carbon cycle. We think that plant growth in the ocean plays an important role regulating atmospheric CO2 concentrations, but that plant growth is limited by nitrogen availability. So then you care about that. Those are the fundamental science questions that ChemOcean is tasked with constraining. So really there hasn't been a ton of work done on submarine groundwater discharge and its role in supporting, both in terms of constraining elemental fluxes, as well as, how it links to biological processes. So we're excited to be working on this and giving this process a little more attention. It's also something that I am working on as part of this GeoTraces group, which is something that NSF Chemical Oceanography has funded. Kirk's gonna get cooked. So we're really interested in trying to bring, GeoTraces is really in the past 20 years provided a completely novel data set of trace metal concentrations in the ocean that weren't possible before because contamination. Because a metal boat, it's hard to get good iron concentration measurements when you're talking sub-nanomolar or iron concentrations. And collecting it, boy, we did this on our recent cruise, and God bless the trace metal chemists, because it is so hard. Like you can't even begin to imagine how hard they work to get their data. Like it is straight crazy.
Well, getting it is easy. Making sure it's real is.
yeah. And so it's super hard. At any rate, so we're trying to bring more work to the Gulf, because especially the Mississippi River, right? Those are that's global scale inputs, and then there's also really significant submarine groundwater discharge on the Yucatan Peninsula. In fact, there's no other freshwater input on the Yucatan other than submarine groundwater discharge. There are no rivers that drain the Yucatan. And so there are globally significant margin fluxes from these sources, and they're draining into a relatively small basin. And so what we really want to do is try and understand the biological, chemical, physical, geological processes that control whether or not these elements make it off the shelf and into the deep Gulf of Mexico or not, and then out into the Atlantic. I'm not sure if any of that made sense.
No, it does. So in terms of data sampling, you mentioned a ship that intakes liquids and stuff. Presumably they have airspace-based sampling type things. But do you have buoys that are set up just to track in real time? How do you do that?
So we don't have buoys. What we do is we sample using a couple different techniques. So we have something called a CTD rosette, which oh yes, look at that. Which is basically a bunch of plastic PVC tubes that have spring-loaded caps on both ends. And those are connected to a computer on the ship, and we can send that down to whatever depth we want. And then on the boat, we tell it what depth to stop. And so we send it down open, and then as we bring what we call the rosette up through the water column, we stop it at different depths and bring water up and collect water. And then there are a bunch of sensors on the bottom of the rosette. And so temperature, salinity, which is really conductivity, pressure, which gets us depth. And there can be oxygen and fluorescence and all kinds of other turbidity and other kinds of sensors as well. And so that's how we collect discrete water samples through the water column. So we'll stop the boat and pick different depths that we want to collect water at. We can also have, like I said, an underway seawater system. So we can get surface water as the boat is steaming. That system is not inherently trace metal clean. And so my trace metal colleagues I'm going to just get destroyed here. This is rough. you guys, let's just watch me get cooked. So anyway, failure. Yeah.
Brutal.
Cheers. So they have something called a towfish that they can tow along the side of the boat, that is hanging off the side of the boat, and swims a couple meters away from the side of the boat, hopefully outside of the wake of all the metal coming off the boat. And then that has like 50 to 100 meters of tubing going into something, a trace metal clean sampling space inside the boat. And they filter that water and collect samples for us all. And it's like a heroic effort.
I mean, so metal sampling, do they do like atomic ionization? How do they find that?
So they have, they've got crazy ICP-MSs and things like that. And then, so they're doing that. And we have people doing iron isotopes and iron speciation and ligands. And so flame, I don't know, Kristen, Buck, are you out there? You want to chime in? Or any of Kristen's people. So yeah, they've got crazy tools to try and do it, and it is extraordinarily difficult, and there are not many people who do it, and especially do it well. And so we have just a really excellent team working with us, working together.
I mean, so in modeling a gigantic problem like this, are you guys just data limited? That you're
yeah, so we're not even trying to model, we're just so we did some experiments, and so it's a lot of correlations, right? And looking for spatial trends and the distributions of phytoplankton, and then the elements themselves. So yeah, and then, this is where, like I said at the beginning, in the open ocean, you kind of have a shot at figuring some of this out because things don't change so quickly. But in the coastal system, where things change on really short timescales, you just need a lot of high resolution data. And this is why it's so important that we're also working with state agencies who have we have these two big cross-shelf cruises, but then to be able to get additional sample sets where we can look over several years and look at how things change in space and time on shorter time scales is going to be really important for linking all this. But we're trying to get constraints on end members coming from the submarine groundwater discharge. And we think that we're going to have some robust end members based on some preliminary data. And so basically, if we have an isotopic composition and we keep seeing the same thing, and there are a bunch of tracers that are consistent with submarine groundwater discharge, then we can be like, okay, this was actually submarine groundwater discharge at this time. This time it was water coming in from the Atlantic. This time it was Mississippi River water coming around the other side. Usually, it's later. We'll talk about that later.
So I don't remember if we had the canned questions last time we were on, but the like unlimited budget, no moral qualms, what experiments would you do?
God.
I mean, what is your ideal data sampling? Like, is it just an army of boats, or do you have like buoys, submarines at different levels? Like, what is your Manhattan?
Thought about that.
Manhattan project purchase. What's your holy grail of
my God
data sampling?
Oh man, that's a great question, and I haven't dreamt that big, at least not recently.
I mean, so we can put
sediment traps everywhere.
Yeah.
And water column profiles. So there are also these profiling floats that are going around, and they have some sensor data. Nutrients, there's no good sensors for nutrient concentrations, and certainly not for isotopes. So you still have to bring discrete samples back to the lab. But I really love sediment trap fluxes. So the sinking, like dead phytoplankton, and measuring that isotopic composition and comparing it with the nutrient isotopic composition, because what goes up is your budget, right? What goes in has to be what comes out. And so I find to be pretty powerful constraints. But deploying a sediment trap sucks. It's really hard. It's really time intensive. Usually you just have it out for a couple days. They also have problems. They don't catch everything they're supposed to catch. And so then you have to compare it with other metrics, and it's hard, but they're still, I think, super valuable and like one of the best things out there. So I would put sediment traps out everywhere. If I could, that would be awesome.
I guess looking at this from a naive perspective, we can put a probe on Mars and do real-time mass spec and sampling of, like, if you had enough budget, you can't build something that does that, or is it?
Yeah, our methods are super labor intensive.
Yeah.
Like they require a lot of people hours. So we haven't deployed something or developed something that could be autonomous.
Yeah. Is that a technical problem or a financial problem? Probably both.
Probably both. Yeah. Really kind of technical. Yeah. So, yeah. And a lot of the things we can't, so organic matter, we can't even measure directly, right? Like, yeah, it's just brutal. So we have to, in oceanography, we have all these operational definitions. So dissolved organic matter is anything that passes through a filter and is reduced, right?
That's interesting.
Because, and so it includes things like amino acids and, peptidoglycan and all kinds of things and pigments and chlorophyll and stuff like that. But, bits of DNA or whatever, but you can't, it's a mixture of everything, right? It's like the challenges that the Mag Lab is dealing with FTICR, right? And these really complicated mixtures.
Yeah.
And so my approach is to look at the bulk pool, but then I have colleagues who work on characterizing the organic matter and then do targeted and untargeted, techniques to try and get at that. And so we're going to be trying to link all that on this project, and then with metals and iron concentrations and biological data as well. So we have people doing molecular work and carbon and nitrogen fixation rates and iron stress markers and phytoplankton, and so RNA and DNA and some transcripts. So really trying to throw everything, not everything at it. We're very intentional choices for what we're analyzing, but a lot of different tools.
I mean, so the flip side of that is the no moral qualms. So, could you drop an ammonia bomb in the ocean and see if it blooms? Like, is that?
Oh yeah, it would.
Thermal generator.
Yeah, yeah. Well, so that basically happened, right? So I don't know if you heard about the Piney Point. So like 2 years ago, almost exactly, right? So this is fun. The phosphogypsum stacks in Tampa Bay. So on the south side of Tampa Bay. So the second largest industry in Florida is actually fertilizer production, right? Because we have all this phosphorus in the rocks. And so there's a lot of fertilizer production. And there's a defunct phosphorus mine called Piney Point on the south side of Tampa Bay. And basically, when they do this mining, they produce there's some radioactive waste, and the state of Florida is obligated just to hold it. They can't do anything with it. But the retention ponds from this old defunct, closed mine started to fail. And what the state decided to do was, rather than have a bunch of toxic waste flow through neighborhoods and drain into Tampa Bay, they started intentionally pumping it into Tampa Bay 2 years ago. And there were ridiculously high concentrations of ammonium and phosphorus in there. And so they absolutely caused a huge phytoplankton bloom. So I got some samples to measure the isotopic composition of nitrogen. My colleagues, so this is all still the state of Florida owns this data. We haven't published this yet, so we can't really talk too much about it. But yes, so we did this experiment in Florida, and yes, you get a phytoplankton bloom.
So do you have the isotopic ratios from what they dumped in?
Yeah.
Oh, that's fun.
Yeah.
So you get to see what actually happened. That's a controlled experiment.
Yes.
Controlled.
And I think we have mass balance. Or at least I think we understand where things went. We don't exactly have mass balance. I take that back.
I mean, does that answer your question though, in terms of like you just need a critical mass of these elements and then it happens?
Oh yeah, absolutely.
It's just a question of what causes that localization or density.
It's really interesting because so one of the theories for 20,000 years ago, atmospheric CO2 concentration was lower, right? And so chemical oceanographers have been trying to figure out why for decades. And there are two mechanisms by which the ocean controls atmospheric CO2 concentration. So one is the biological pump, which is just phytoplankton growth, right? It's a fancy way of saying phytoplankton, plant growth in the ocean. And the other is called the solubility pump. So just the degree to which gases are soluble in the ocean, right? So strictly physical controls, temperature and salinity. And there are indications that during the last half of atmospheric CO2 drawdown during the last glacial cycle, that there was a stronger biological pump in the sub-Antarctic. And somebody named John Martin had a hypothesis around 1988 or 89 that basically when you have a colder climate, it's drier and windier and dustier. And he hypothesized there was more atmospheric dust deposition to the Southern Ocean, and that fertilized the biological pump, which right now is not living up to its full capacity. There's a bunch of phytoplankton that are not eating the macronutrients, nitrate and phosphate. And so a lot of people have worked there to try and figure out why. It's like children not living up to their full potential, right? Like, why aren't you getting good grades? Like, you could be working harder and pulling more.
That was a brutal round of nonstop, like, poor people. Jeez. Congratulations.
Yeah.
Victory there.
Did I win? I'm not even thinking.
Yeah, no, you did.
Gold stars for me. Yeah.
Watching, that was a lot.
I tell you. So
have you beaten this game before?
I've beaten this hand.
Yeah.
We'll see if I can do it without my regular controller. But I have beat the hand.
Yeah.
But man, that like pinball when I was falling down that thing, like that's tough stuff.
But that's like a bonus level, right? That's
right.
Yeah.
Yeah. So, anyways, I gotta not be smashed.
No worries. It's on the hand. Do you want more, or do you want to try the can?
I'm enjoying whatever. I still have some in here, so I can keep going. But so at any rate, we think that the Southern Ocean was fertilized by increased atmospheric dust deposition. 40 to 20,000 years ago, and that pulled atmospheric CO2 down to about 180 parts per million. The pre-industrial atmospheric CO2 concentrations are about 280. And right now, of course, we're like at 420 or 425 or something like that. Mm-hmm. But what's interesting is a lot of different groups have tried to go out and fertilize the ocean with iron in the Southern Ocean and other regions that we call high nutrient, low chlorophyll, which is basically regions that aren't living up to their potential. They have plenty of nitrate and phosphate in the surface ocean, but the phytoplankton aren't eating it. And we're like, well, why? Why are you being lazy? And we think they're usually iron limited. And so what's interesting, though, is that, I think this is a really important point, and a lot of people often think you can outsmart biology, but I don't really think we can. And so what happens is people add iron, but they don't actually end up increasing export production. So they can make phytoplankton grow, but if the phytoplankton don't sink out of the surface ocean, they're not moving that carbon out of the communication with the atmosphere. So the surface ocean is constantly in communication. It's in equilibrium with the surface ocean, or with the atmosphere. And so unless your phytoplankton sink to the bottom of the ocean, they're not moving carbon away from the atmosphere. And so people don't really understand why we aren't, when you add iron and like John Martin famously said, if you give me a barge, I'll give you the next ice age, a barge full of iron shavings, basically. Mm-hmm. And people have basically been trying to do that, and they can't make it work. And so then the question is why?
I was going to say, we've sunk enough ships to test this, right?
No, no, more iron. And so I think it's basically that the phytoplankton have optimized for the current conditions, right? And we can't outsmart them. And it takes some time for phytoplankton to evolve and change and, optimize for different conditions. So I think it's, there's still work being done on this, and people are going to keep doing these experiments where they try and fertilize and pull CO2 out of the atmosphere. But right now, we're not succeeding in that effort.
So I guess a lot of the stuff we've discussed is this top-down approach, trying to understand systems that exist. Are people doing like terrarium style, like swimming pools of like just add crap and see what grows? Is that a viable strategy?
That. Yeah, it happens in everybody's swimming pool in Florida this time of year. There's actually atmospheric dust deposition from Africa, and so we see phytoplankton blooms as soon as those start coming over.
That's crazy.
And so do my colleagues. So yes and no. You can't really bring back seawater to the lab and then just manipulate it. People do those manipulative experiments at sea, and there's a long history of that. It's hard. It's real hard. There's these things called bottle effects, right? Where you sort of expect the longer a lot of people don't want to keep things in a bottle, no matter how big it is, for too long, because it just stops acting like nature and you're not sure how much. Yeah. So people do, but people have differing degrees of enthusiasm for those kind of experiments. Usually, if you're trying to measure carbon or nitrogen fixation rates and you're doing incubations, you usually don't let those run for more than 24 hours. But some of my French colleagues have done some huge mesocosm experiments where they're like, what was it, 15 meters deep and a couple meters across, and they had scuba divers and stuff like that. So I have a paper with my French colleagues on some of those. And so, yeah, they do some really large scale mesocosms, and they definitely saw a shift in community composition. I think they let those run for at least 2, I think maybe 3 weeks. So people will do all kinds of things.
I mean, so that's the middle scale, I guess. Like, is there a petri dish equivalent of growing phytoplankton and just mass balance, like feeding it amino acids, nitrogen? Like, do we know how those behave?
Yeah, so we do. So you can do culture studies, right, in the lab, and then, but those are one-off, right? And you're like, okay, I'm going to feed you an amino acid and see if you'll eat it. And so my colleague Sven Kranz has been doing some of that with the K brevis, or we were going to try and do some of that. And he's moving, but we're still going to try and do these experiments. But at any rate, and so people do try and do that, but then it's like, okay, I learned that about one phytoplankton. Yeah.
I mean, does that give you insights into like the critical role of iron and how much, and like, I mean, does it? Yeah.
Yes. And so Sven Kranz and I are also doing some experiments with what we hope are representative species from the Southern Ocean. So he's grown some diatoms in particular in the lab, and also a coccolith, E-hux. And so we're doing some manipulative experiments where he's varying iron and light, and we're seeing how that changes how they take up nitrate and the isotopic composition, or specifically the isotopic fractionation of the nitrate. Because that actually is the link to the paleoceanographic record. So the link for how we think we know that the biological pump was stronger in the past is actually through nitrogen isotopes. So we actually think that they were consuming more nitrate in the surface ocean. And so the link is nitrogen isotopes, but to get to that link, we have to have a robust understanding of the isotope effect, or the discrimination of phytoplankton against the isotopically heavy form of nitrate. And so he and I are actually testing that. Well, he's testing that in the lab by growing the phytoplankton, and I'm just measuring the nitrate that he gives me.
That's awesome. I like the isotopic studies. Those are really fun. By the way, did you beat this?
I don't think so. Did I? I don't know. Did I?
Press start.
Let's call it. Let's call it I won. I don't think I did.
Does anyone know Smash Brothers?
I just do what my kids tell me.
You beat the hand.
I'm just there to be beaten by them because I don't care, and they get upset and they want to win.
I guess you're fighting Kirby now. Is that?
I don't know who I'm fighting, but yeah. So I don't really care if I win or lose, and they get excited.
What is this one called? Don't know my Pokemon. But yeah, the isotopic studies, for anyone not familiar, but basically you have heavy atoms, and these just exist. These exist in ambient conditions and things like that, but biological systems process those heavy atoms differently. Not only that, just like the distillation of, you take heavy water, you can separate heavy from light just based on heating it.
What's in the glaciers has a lighter isotopic composition. So, but biology discriminates. It's very rude. So enzymes discriminate against the heavy isotope. It takes more energy to break those bonds.
In fact, if you drink, it's something like 5 liters of heavy water, you'll die because your biology cannot process it at the rate necessary. Yeah, it's just it slows down the kinetics enough that you can't process.
Wow.
Yeah.
Wow, good to know.
Yeah, don't drink heavy water.
We don't have any in the lab or anything like that.
It's expensive.
It's hard to get your hands on 5 liters of that.
But it's like, basically, if you screw up all the hydrogen bonding and whatnot, it's, everything goes haywire.
Wow.
Which is cool. That's how they know the temperature, 500,000 years ago from ice core samples. And they can have water ratios from that, which is really, really cool process. But
I didn't know that the heavy isotopes messed up hydrogen bonding. Huh.
Yeah, if you have deuterated water, it basically slows down that vibrational mode. And we actually use that to figure out, is this hydrogen atom involved in a reaction? Because if the reaction slows down, we can directly correlate it with that event.
That's cute.
Yeah, no, it's really cool. And I'm assuming that's what the plants are doing, basically, but it's a very selective version of that.
I hear it's about 0 point energy, which I hope you could tell me. And that's your job as the actual chemist in the room.
It's about the 0 point energy.
Go ahead and give a lecture on that. Yeah.
I was going to say activation barriers.
That's,
So I don't know what just happened. You
I don't
lost that
know.
round and then it was over.
I don't understand,
Like,
Ken, but what do you want me to
the
play?
Special. What do you want to play? What do you in the mood for? More of this or play? We have Mario World, Mario Kart. We have Tetris. What do you want to do?
Oh, let's try some Tetris.
Tetris.
That's probably really boring for people to watch. But
That's
okay.
Everyone knows and loves Tetris.
Okay.
Around to that controller.
Thank God.
We have 3 generations of Nintendo controllers in front of Angie right now.
Yeah. Something
Progressive
more like
tracing complexity.
what I'm used to using. All right. Okay. Okay, now we're talking, Ken.
And so, I mean, so you mentioned like there are people that are doing these experiments, just like dumping stuff in the. What does it take to get approval to do that? Because that's like human experimentation borderline.
Yeah. I think it depends where you're going. So I haven't been involved. There were some people who tried to ask me to be involved at some point, but it was not really super.
Just like put on some black masks and dump shit in the ocean.
No, I think it's more complicated than that.
Yeah.
But yeah, that is not how I have been spending my time, is trying to do that stuff.
I mean, it's intriguing because like that's what we want to do, right? Like you ask a sociologist studying human behavior if you could just track people, but you're doing it on an ocean scale, which
Right.
is intense.
Correct.
I don't know how to think about that.
Yeah. You hope that you're right. And some people have more hubris than others.
Well, I mean, so as a photochemist, I'm curious about, I mean, these are photosynthetic organisms, right? Like,
Yeah.
you could just start shining a strobe light at the Gulf of Mexico and, like, induce it? Like, is that...
Well, so that's what Sven Kranz is doing in the lab. And so I call it the phytoplankton discotheque. And so I think it's great what he's doing because a lot of phytoplankton cultures have been grown under what I would call not environmentally representative conditions. And so they'll basically grow phytoplankton under fluorescent lights. And then under a constant, like 24 hours light. But that's not what the real world is
cycles.
like. Yeah, exactly. And so what Sven did is he built with Arduino boards and Raspberry Pis. He built a system to grow phytoplankton where he could program the wavelengths of light to be representative of what they actually experience. And then the other thing that he did was that he varies the intensity in space and time so that it's like a cloudy day.
Yeah.
And so he's growing the phytoplankton under all these different conditions. And then if you look at it, like somehow, I don't know, it looks like a discotheque or what I would imagine a European discotheque. I don't know that I've been to one. But I call it the phytoplankton discotheque. I don't think he likes that name very much, but it's kind of awesome. Whoops.
So the 24-hour day strategy, that was just to speed up growth, or that was just, it was easier that way?
It's easier. Like people didn't have a controller, you know, and people were
Yeah.
doing this like 40 years ago, you know, and it was just like, well, turn the lights on and let them go.
It's much easier to simulate the sun now. So that makes
Right.
sense.
Yeah. So there's a lot easier, cheaper, you know, controls on all this stuff and electronics and whatnot.
I mean, it's intriguing. So thinking about that from a human perspective, obviously we need sleep at night to recover. And like, I mean, every emergent phenomena that people study, they requires some kind of flux of energy, not a continuous supply.
Right.
And so is he seeing that does matter?
Yeah. So what's really interesting is that, you know, so he's varying light and iron stress. So he's subjecting them to varying ratios and sort of concentrations of iron and light stress. And what's interesting is that basically he knows, I haven't seen all his data and he can certainly speak to it much. I'm a geochemist. He is a phytoplankton physiologist.
Yeah.
But my understanding is that phytoplankton have developed a lot of strategies to deal with light stress, but it is much harder to deal with iron stress because there are enzymes that are required, right? And you can't, after some point, you can't substitute your way out of it. And so they're just limited. But there are a lot of tricks that phytoplankton can play to deal with light stress. So phytoplankton have a lot more tools to deal with light stress than they do to deal with iron stress, is my understanding.
Oh, that's fun. One of the things I'm lacking in chemistry is biochemistry, but I'm curious about like photosynthesis and the driven processes of light.
Yeah.
So that's fun.
It's really interesting. It's super complicated. And I'm always like, oh my God, I need to read 500 more papers. Give me the grim notes. But yeah, I never feel like I know enough. So.
I think that's all of us, unfortunately.
All the time. Oh my God.
Yeah. We have no time to do it. So.
Correct.
Yeah.
That is true. Oh, this is a little tough here.
When was the last time you played Tetris?
I don't know. Months ago? Many months ago.
Yeah.
Yeah.
But it hasn't been 30 years or something.
No.
It's fairly recent.
I mean, within the past year, I probably played it a couple times, but not a lot. It's not something I spend a lot of time. So we'll try again here.
Believe it or not, there was a renaissance in like Tetris gameplay in the last 3 years or so.
Really?
Yeah. No,
Is it because we all played it when we were little and now we're doing it again?
No, there's actually, they came up with a new technique and it's called rolling. So you're fundamentally limited on how fast you can push the button. Right?
Yeah.
Like if your thumb is going like this, there's a certain rate you can do. What they figured out is you can hold your thumb on the button and roll your fingers underneath it.
Oh my
God. So you can like tap your fingers so you can get 4 hits in the time it would take you 1 press.
Oh my gosh.
And so it's called, I think it's called rolling or something like that.
Wow.
But that's how they play now. They hold it and they tap underneath to get the right number. And it's basically the number of fingers is the number of movements you have.
Oh my goodness.
And so they've almost beaten Tetris. Like it doesn't have an end, but like if you
I
don't
beat it
lose,
once in like, I don't know, junior high or something on my Game Boy.
Yeah,
I beat it once.
You can get to a certain number, but like it lets you keep going right beyond
wow.
the end of the game, and that's what they do. They just try to maximize scores and get there as fast as possible. But they've gotten to the point where they can't lose anymore.
Wow, that's amazing.
Yeah.
Yeah, no, that's not gonna be how I'm spending my time these days. But a few too many commitments.
Yeah.
So yeah.
No, it's fun that after 30 some years, there's something new in this game,
Right.
which is pretty breathtaking.
I haven't seen it. Wasn't there a movie too on Netflix or something like that?
It's either Amazon Prime or Apple Plus, the Tetris like origin of the game. I haven't actually seen it yet.
Yeah, we haven't watched it. I think we saw advertisements for it.
Yeah.
We're watching Ted Lasso.
I did watch the Mario Brothers movie.
Oh, you did? How was that?
It was pretty fun. We
Yeah.
brought the daughters to it, and it's a mix of nostalgia and like, yeah, it was
Silliness.
It was a fun portrayal. It's not like it's a profound plot or anything. It's
Really?
just like, hey, look at that! I recognize a bullet bill, and like, yeah, I had that in my childhood. So yeah, it's pretty
Excellent.
fun. It's worth the trip.
Nice. Okay, good to know.
I guess the comparison is the 1990s Super Mario Brothers. Do you remember this one? John Leguizamo and what is his name from Who Framed Roger Rabbit?
Oh,
Yeah.
wow, that guy.
Yep.
Okay, yeah, nope, haven't seen that
Bob
one.
Hoskins.
Yeah.
I think.
Oh,
yeah.
that was a good pull from the memory. That was a deep
Yeah.
cut there again.
I watch a lot of movies.
Got some little kids. Yeah.
So speaking of movies, one of our other favorite questions. I mean, movies or TV shows that you've seen that like they get your discipline, your expertise, your profession right. Which ones get it wrong?
I haven't seen any that I think are closely related at all.
No, not Deep Blue Sea with sharks. No.
I haven't watched that.
It's
But
great.
I think, when was it? My oldest, was it over winter break? It was just before I went to see, maybe it was over, I don't remember. But we watched Jaws. She hadn't seen Jaws. So we
Yeah.
watched Jaws, which is not like what we do at all. Although Richard Dreyfuss, like trying to be an academic, maybe. Okay.
Academic-ish.
Maybe. Yeah, ish, kind of obnoxious. I guess he was also from an independently wealthy, he wasn't really an academic, right? He was an independently wealthy.
He was a hobbyist.
Right. Which is great. Yeah, so that was the thing most related that I've watched most recently.
Yeah. And did they get anything right? Did they?
Well, I definitely don't do the charismatic megafauna.
Yeah.
So trichodesmium is my version of charismatic microflora.
Yeah.
But it was fun to watch.
But in terms of the sailing, is there anything? I mean, you've learned a lot about sailing over the years.
I mean, some. So we aren't out on. Yeah. So our boats are
Huge.
So bigger than that. The one that Jaws was out on, although sometimes not that much bigger. But yeah, you want to make sure the engine doesn't blow. Don't do that. Yeah. But yeah, that's so there are winches and things like that we use to haul our gear over the sides, but no shark cages, nothing.
We
don't use those. No. I don't, and I'm not aware of other people who do. You need a whole lot of permits to make that kind of thing happen on NSF funded research, I think. So, and that's
fair.
Not my jam. Yeah. So
that's fair. But we're an hour in. Anyone just joining us, our guest today is Dr. Angela Knapp. She's an expert in oceanography, and particularly the marine nitrogen cycle and phytoplankton and the processes that dictate algae blooms and things like that, or trying to understand them. She regularly goes out on boats and measures a whole lot of things, trying to understand what variables dictate biological processes within the ocean. So yeah, if you guys have any questions, throw them in chat. We're happy to answer them while we play a little bit of Tetris. Which I gotta change the name. I should look up if you beat Super Smash Brothers.
Either way. This is kind of my life.
We'll count it as a victory.
Yeah, sounds good.
I mean, so you're how often do you go out then? These trips?
Yeah, so good question. So I don't go out that often unless it's my own cruise, because I have a lot of colleagues who do experiments at sea, or like my trace metal colleagues, God bless them, it's super hard to collect their samples. Mine are not that hard to collect. And so usually what I do is get seawater and I put it in the freezer and do most of my analyses at home. So it's not really a great use of my time to be at sea unless it's my cruise that I'm running. And so this time so I'm the lead PI and the chief scientist on these cruises, but it was very much a team effort. It was a very complicated sampling strategy that we had. But it actually ended up being good that I was the chief scientist, because mostly my trace metal colleagues were collecting samples for as many people as possible so that we could have one sample stream, right? And so that everything was collected one way, so you don't have to guess like, oh, is my sample different than theirs because something got contaminated or whatever. They all came from the same thing, which is really lovely. But then what that meant was that I wasn't like processing samples myself, and so that meant that I could sort of tell the boat where we needed to go and take care of all the logistics of like, okay, this is the next station we're going to, this is where we're headed, because there's a lot of that has to get done. And so the fact that I was able to do that while my trace metal colleagues collected samples for me was definitely a luxury for me, but meant that, I just, talking in Tetris.
Yeah. I mean, so you're collecting samples, you bring them home, that lasts you for like a year, or like how long?
Sort of depends, on how the mass spec is working and how many samples we have to analyze. So yeah, it all depends. But yeah, we try and analyze them, say, within a year of sample collection, and everybody else is too, and everybody else has very fussy instruments. And so, getting everybody's analyses to line up and all get done is a thing.
So like once a year is not unreasonable for trips and data collection?
Yeah, it just sort of depends. So right now, some of my colleagues they're going to go out and get more on the West Florida shelf, more of these samples, just because, like I said earlier, conditions change out there so quickly that it's important to get coverage. And so, but those are usually somewhat smaller, well, much smaller sample sets. And so you can get through those a little bit faster. And it depends what the funding you have to analyze those samples is like, and how many of the things you're working on.
So how does that work practically in your community in terms of like you gotta get teaching release built into your grants, or how do you get that time?
Yeah, no. So it all depends on what institution you're at.
Yeah.
So I have been teaching this semester and being a chief scientist, which I'm real tired.
Yeah. Wait, does that mean Zoom teaching during that travel interval?
So how many classes? I think I missed 5.
Okay. So you just
lectures,
take that window
something like that. So Sven covered 1 lecture for me. My grad student covered 2. He got to talk. I'm doing a grad seminar this semester, and so we're reading papers. And so my grad student presented 2 of his papers, or 1 of his and 1 of mine. And then 2 class periods. They had for writing, a writing assignment, so they got time to go work. Like if you're at a meeting, you miss 2 classes.
I mean, even that, like 5 isn't through a semester is not bad, because that's I mean, sick kids will do that, right? Like
correct. Yes, they will. So 5 was okay, but if it gets to be more than that, it gets cumbersome. So some of my colleagues double up their teaching assignments. So different institutions sort of do it differently. And then in our department, we have colleagues who are modelers and don't go to sea, or people who are have most of their field work focused in the summer, and so the oceanographers kind of catch as you can and figure out what they need to do, but we usually make it work. We're going fast. Oh no.
And those are long trips. Speaking of which, we should do a prediction, because I've been sitting on these too long.
Okay. All right.
This one is, in terms of trips, is kind of fun.
Okay, here we go. Yeah, this is good.
Anyone that's not familiar with the channel, we have predictions on the stream. We don't do predictions related to gaming, or at least usually not related to gaming. We do science-related questions. But yeah, if you're not following us, click the follow button. Your 300 standard internet units, you can bet them on the answer to this question, depending on how confident you are. This is a research question related to Angie's research. So if you're not familiar with predictions, at the top of your chat, you'll see a button that says predict. You can click on that predict button and bet on this question. The question is, what's the fastest time from Tampa Bay to the Deep Gulf? And so you're talking about
if you're on a research boat. Yeah. How long does it take you to get across the West Florida shelf into deep waters?
Not a speedboat. Just like a standard, think a Jaws boat, slightly bigger. About that speed. How long does it take you to go from Tampa Bay to the Deep Gulf? And so the Florida, there's like it's called the shelf, right? It's just like this, I don't know, it's like a massive sandbar. How do you think about that?
Yeah, so 20,000 during the last glacial maximum 20,000 years ago, that was all exposed, and that was all part of Florida. So Florida was twice as wide as it currently is.
And so you have to drive your boat over that to get to the Deep Gulf. And the question here is, how long does it take to make that sail? Like, is it 5 hours or is it 1 day?
Yeah.
How long does it take to get over that the Florida shelf?
And we'll find out if anybody from the February-March cruise is tuned in, because they will know. They are going to be like, "Oh, I knew that answer." Yeah.
So make your predictions now. You guys have about a minute left. I don't think you can Google this one, but good luck. Chat GPT, see if they know it. But
I'm one step ahead.
You're in a standard research boat. How long does it take you to get from Tampa Bay to the Deep Gulf, as in past the Florida shelf?
Slope break. Yeah.
How long does that take? Is it 5 hours or 1 day? It's intriguing, because to drive from north to south in Florida is...
Because I think I have the mileage, right? It's like, what did I say in there? It's like 250 kilometers or 155 miles.
Yep.
So think about how long that takes, to drive versus if you're in a research boat.
It's time scales I just don't think about, like in terms
Oh my
of
God.
sailing distance.
So these are the things that chemical oceanographers have to deal with when we're writing NSF budgets. We have to map all this stuff out. And
Yeah.
so when I'm telling you, we have it harder.
Well, that's why I was curious about the
added degrees of
timing, because
difficulty.
let's say your boat breaks down and you missed 3 weeks of class, because that happens.
It happens. And boy, does it happen these days. So if any of my Atlantic Explorer friends are out there, so we love them because they brought us a boat that worked. And there are all these horror stories out there right now about, we could spend the rest of that evening talking about how things have gone wrong for colleagues' cruises, and it's brutal. And we just got super lucky. And we also had people who made sure our boat was really prepared and ready to go for us. And so we're very grateful. So if they're out there, thank you.
All right, Angie, how long does it take to go 155 miles?
About a day.
About a day. That is a long journey.
It's nice transit. It gets you ready if you can. If you don't have to stop along the way, it gives you some good time to get your sea legs under you. Stop barfing.
Brutal. Oh, Scuzzbot redeemed. Take a drink. Do you need more, or are you good?
Sure.
You're good?
Yeah.
All right. Let's get this guy.
Yeah.
Speaking of which, what are we drinking tonight?
We are drinking gin and tonics, and if anybody was at sea with me, they will appreciate that. Although boats are totally dry, like we do not have any alcohol out there, but when you get back on land,
Yeah.
you
But
get to
that mandated?
relax a little
That's,
bit. Yeah, that's very strict NSF policy. So they can do drug and alcohol tests. So there is absolutely, it's completely dry. There is nothing. There is no, although it's interesting because again, the French boats, and I have South African colleagues who have the world's newest icebreaker. They have white tablecloth service at dinner, and they have serve you wine. And I hear that there are bars on these things. I have never been on an international boat. I've only been on American boats, and I've never experienced any of this. So, sounds amazing. Although, honestly, I think it's a good policy because we deal with a lot of very heavy equipment, and it's already pretty scary. And well, it can be if you're not careful or if you got people who aren't paying attention. I don't want anybody who's tipsy in any way, shape, or form dealing with any of that heavy equipment. So,
So Scuzzbot, cheers. Welcome back to the stream. Thank you for joining us. Which gin? I don't remember.
Oh, you
the
picked
name
it
of
out.
I did.
So,
I
but
have
it's
it
good.
in the other room. All right, I'm going to grab it.
Yeah.
I'll bring it back.
All right.
Scuzzbot wants to know.
All right. People are just going to watch me play.
I should have given you a question.
It's okay. Hi, everybody. See if I can clean anything up here. Nope. I'm trying to clean up tiles, Ken, and it's not happening.
All right, so we're drinking Plymouth gin.
It's good. I don't think I've had it before.
For those of you that don't know, I ask every guest what they want to drink. And then when they tell me, I go to Google and I say, what is the best gin for a gin and tonic? And then I buy something that's on that list.
What a good host.
So I've acquired a semi-large collection of those. So that's kind of fun. But also last week, so Lance Cooley from Mechanical Engineering, he likes to age his own whiskey. So he brought over this like 400 milliliter bottle of his own aged whiskey, and he downed it.
So I could, So before Christmas, right, we had that hard freeze in Tallahassee, and I'm making some limoncello. And I haven't. Oh, we're gonna lose this Tetris round. But I have limoncello working in my pantry, but I have not dealt with it. It's just been like lemon peel and vodka right now is what's going on. So I think I have some lemon simple syrup in my freezer. I think all I need to do is mix it. But at any rate,
well, when you're back in 2 years,
that's right. Bust out the limoncello. Yeah.
But yeah, I don't usually drink gin and tonic, but yeah, it's good. We have fresh limes, ladies and gentlemen.
There you go. Not from the yard. They're not from your yard.
No, absolutely not. They're from Publix or Whole Foods. I don't remember where we got them from.
That's all right. Our lime tree, I don't think survived the hard freeze.
If I remember correctly, last time you were on, you just got solar cells, or you were purchasing them.
We did, yeah. So we got solar
that going?
panels on last summer. Great. So we haven't had them on for a full year yet, but and my app that tells me I
you can track it. That's fun.
it's kind of satisfying.
Yeah.
So I think we were predicted to get 80% of our energy covered, and I think we've been doing at least that, but I haven't checked basically since I've been at sea. So
You should graph your solar photocurrent versus algae blooms. just see. Just see if there's any correlation at all.
Exactly. So
a little far away from Tampa, but so it goes.
that's right. Yeah. So we've been really happy with them. They've been great. And we're going to try and get the pollen rinsed. I think the roof is supposed to be washed and get all that Tallahassee pollen off.
That's a non-trivial drop in efficiency.
Yeah, I haven't seen it because it has happened since I've been at sea and I have just lost the plot. But yeah.
No, that's fun. I would be watching that data as they the day after they clean it, you have a 40% increase.
Yeah. No, it was real fun to watch right when because they got installed, I want to say August last year. And so sort of at the end of the summer. And but yeah, they've been great and yay for us.
Yeah. So. I'm jealous. We gotta get some solar panels. We have too many giant trees around us, unfortunately.
Yeah, we have some big trees, but we have good southern exposure. So our yard worked out just right to do it. So that was convenient. And then we ended up getting an even better tax break than we expected, right? Because Biden extended the...
Does that still exist? Because we've been debating.
I don't know what the latest is. All I know is we got more back than we expected, which is fun. Yeah.
It's pretty fun. All right, anyone just joining us? Our guest today is Dr. Angela Knapp. She's an expert in oceanography and marine nitrogen cycles and phytoplankton and trichodesmium. She's happy to answer questions about any of those things while she's playing Tetris. And we're going to say you did beat Super Smash
Let's
Brothers.
say that. I love that answer.
Yeah. That's perfect. It's not like there's YouTube evidence of this.
Oh my God. I can only do so many things.
Oh man. Well, I'll just say it's fake. It's AI generated.
Yeah, perfect. Thank you. I appreciate
At some point I realized there's enough footage of me on stream now that someone could fake me and speaking, and
that's terrifying.
Isn't it?
It'll be your kids when they're teenagers. Be like, my dad totally gave me permission to do that. Check it out. That's what's coming your way again.
There's a huge record, and oh man, it's terrifying. I love and hate big data,
Yeah, I am not. Yeah, I don't like future dystopia, so I just try
pretend it's not happening.
Yeah, that's my strategy.
I'm sure this will all work out.
It's like me answering science questions and playing video games and drinking a gin and tonic.
Yeah, that's fair.
No problem.
I'm gonna try one.
You're going for okay.
Yep. All right, so those of you who might have seen this massive uptick in hard seltzers and ranch waters and stuff like that, but accompanying that is a non-trivial portion of canned cocktail drinks. And so when Angie asked for gin and tonic, I got gin and tonic water, but also I found cans that are gin and tonic. So we're gonna see how the Bombay Sapphire gin and tonic tastes. Are you curious? Have you tried?
I have tried one. I think my brother gave me one once.
Yeah.
It's all right. It does the job for sure.
I mean, it's a much lower alcohol content. It's 5 to 10% rather than.
Well, where's the fun in that? Yeah. What do you think?
It's not bad.
It's not
bad. I
bad.
don't have lime. Captures something.
You got lime. Well, you just cram one in there, Ken. Just make it work.
I'll just hold the lime in my mouth.
Be an oceanographer. Make it work.
Oh, man.
Yeah.
That's brutal. But yeah, if anyone has questions, throw them in chat. Angie's happy to answer them while playing Tetris. Because, it's not hard.
Why not? Try me. It's not the hardest thing I've done all day.
Ooh. Dr. Pinner? Dr. Knapp, can you explain why some CO2 in the ocean eventually turns into limestone and other CO2 can become a greenhouse gas?
Okay. So, The thing that controls whether or not you precipitate carbonate minerals in the ocean so there are several factors. So pressure, salinity, temperature, so all of that. So one of the things that's different about gases that they're actually more soluble in cold water than warm water, right? And so one way to think about this, so coral reefs, right, are calcium carbonate that's been precipitated. And so calcium carbonate likes to precipitate in warm, shallow waters. So the way oceanographers think about this is you can sort of think of carbonate minerals precipitating in the shallow places. It's like snow forming in the ocean, but it's shallow, right? And so if you get confused, what I tell my students when we talk about this in Chem Ocean of where you expect to find carbonate minerals precipitating, think about where you want to go snorkeling. And it's going to be the warm, low latitudes, right? You're not going to go snorkeling in the Southern Ocean. There's not a lot of carbonate mineral precipitation there. In fact, the Southern Ocean takes up a lot of CO2 from the atmosphere. So that's one of the main CO2 sinks for the solubility pump. So that's the other mechanism by which the ocean regulates atmospheric CO2. So there's phytoplankton that just eat CO2 and turn it into biomass. And then it's just the dissolution of CO2 gas in the ocean. And so there's a lot of CO2 gas dissolution in the Southern Ocean, but mineral precipitation in the low latitudes, in shallow waters, and warm waters. I hope that answers the question.
Yeah. Well, that's fun. That's like general chemistry to equilibrium and temperature and solubility. And yeah, I teach all this stuff.
Hey, there you go. Maybe if they take your class and mine, they'll get it.
I mean, it's not that there's a certain like what I hate about traditional teaching of general chemistry too is a lot of plug and chug math, right? Like here's the temperature, how much solubility is there, and like I really try to emphasize the relationships between things, right? Temperature goes up, solubility decreases, at least with a gas and a liquid and things like that.
Yeah.
But like the CO2, I have a lot of fun talking about the calcium carbonate and like the limestone basin. And because it's a very cool equilibrium because there's CO2 gas versus CO2 in aqueous solution, right? So that's a phase equilibrium, right? And then there's a CO2 plus H2O making carbonic acid, which is a chemical equilibrium. It's basically a Lewis acid base K formation equilibrium constant. And then you take that carbonic acid and that has an acid base equilibrium where it generates, H plus, and then you have the acidification of the ocean and things like that.
Exactly. This is ocean acidification.
Yeah, and there's this really cool, and I'm not going to try to dig it up right now, but it's basically, it has, in general chemistry too, we cover, Kas, Kbs, Kws, Ksps, Kfs, Kds, and there are all these different equilibrium constants, right? But all of them are involved in that cycle, right? Because ocean acidification causes, calcium carbonates to dissolve because the Ksp is influenced by the acidity leaching out carbonates, and it's a beautiful multi-equilibrium problem.
You cover this more comprehensively than we do. So we talk about it very conceptually. So when I do Chemosh, it's got both undergraduate and graduate students in it. And so yes, we don't go through all those hardcore equilibrium constants, but I'm glad that you do. Send them to your class, and I will ask them now for undergraduates if they've taken your class. I will expect that they actually know this. They should be getting these questions right, because I'm doing a much gentler version of this.
If you ever want to guest lecture on that topic,
oh my God, you're on. I am totally having you every year from now on.
Equilibrium.
It's on. Yes. Fabulous.
I got to show you my favorite slide from that.
Yeah.
Where is it? Sorry, everyone. I also talk about kidney stones, which is the same thing.
Oh, God. It's fun. Yeah.
On a different scale. Oh, man. Where is it? Oh, yeah. We'll talk about eggshells, dying eggshells. You do a vinegar treatment at a time for the same reason.
Okay.
Oh.
We also get into aragonite versus calcite. Do you go into aragonite versus calcite?
No, I don't go that far.
Okay.
All right.
Oh, look at, yes,
That's
Ken.
a Chateliers principle.
There
right
you
there.
go. Oh,
All right,
nice.
can
This
you
is,
pause yours. I'll make it so
we'll
they
try.
can see this.
Yes.
All right, everyone. I'm going to show
some gin
you
and tonic.
what we talk about.
We're going to totally launch into
There you
chemo
go.
right here. All right,
No,
here we
so-
go.
So yeah, there's a aqueous solubility here, right? CO2 to CO2, CO2 plus H2O make carbonic acid. There's the Ka equilibrium. You have H plus and OH minus. That's a Kw equilibrium. OH and HCO3. That's a Kb equilibrium. And then this is the Ksp equilibrium. And so these arrows are just like atmospheric CO2 goes up. This goes up. It shifts this way. This goes up. This goes up. That goes down. This shifts. This shifts. It goes that way. You dissolve.
Oh my
sea
God.
life.
Look at that. Yes.
Isn't that fun?
This is the ocean acidification. That's right. So you liberate all these protons, and protons lowers the pH. So yeah, we talk about this in ChemOcean all the time. And what's funny is that, increasing fossil fuel consumption and burning of, production of CO2 in the atmosphere is what drives ocean acidification. It's always funny to me that people fight, or there's some resistance to the idea of global warming because of loading the atmosphere with CO2, but no one's really fighting ocean acidification, but they're two sides of the same coin. Like, they're all the same thing,
Here's
friends.
the other one. So I talk
See.
about the coral reef
There you
bleaching,
go.
but the other
You
one
just
is
want to be in our department. Look at you. You just,
the other one is sinkholes.
Oh,
and the
correct.
limestone.
well, and that's where we get our submarine groundwater distributor going.
So yeah, no, I'm happy to talk about it. I
Look
love
at
this
this.
subject matter.
We're just going to pull you into UIS. This will be great. Chem,
Yeah, I mean, I don't study it; I just teach it. But like,
Good
if you have
enough.
things that glow, I'm all about
check.
it.
Oh, well, we've got phytoplankton and they bioluminesce. Actually, I have questions about photochemistry.
Oh, let's do it.
Yeah, see, we're
On
going to turn
stream.
chem into an oceanographer. It's going to
I'm
be good.
happy to do it. You get me out on a boat for a week?
Oh,
Sure, let's do
actually,
Let's do it.
You want to come in June and July? We have an opening for a male. We have one male birth.
It's gendered. It's based on
Yeah, so
occupancy?
you're correct.
Wow.
Yes. So Unal's boats and I think NOAA boats.
Apologize
can
for
have
causing
mixed-
that death.
That's right. I think it was on the way. Yeah. So you have to. No mixed gender rooms.
Oh, okay.
And we have room for one more guy, but we're hoping for experienced sea hands.
Yeah, I
We've
was
got
gonna
a
say,
lot of work to
I'm
do.
a prop in
Have
that.
you been to Steve?
No.
No, you don't know how barftastic you are.
No, I don't actually.
Yeah.
I'm very curious.
Pretty much everyone is barftastic, and everybody just works to figure out their seasickness meds. And yeah, that's just the truth. Although June and July in the Gulf, usually, I'll say this and then we'll get tossed the whole time, but usually is pretty flat and calm. We've had very nice conditions in the past. It's been very calm, but again, I say that now, which is pretty much guaranteed to jinx it.
I can't remember which guest it was. I had a guest that was like a casual sailor. And
Oh.
they did the trip across the Gulf of Mexico, which is like, it's like a month or something. Like it's non-trivial.
Yeah, it's big. Turns out the Gulf is big.
It might have been Scott Stagg actually that did that just for fun in the summer. Which is kind of intriguing.
Yeah.
But yeah, no, I'm all about light chemistry, and equilibrium is my favorite thing to teach in Gen Chem. So
All right. Well, guess what? I'm gonna call up Ken for ChemOcean every
Whoa.
fall now. Be like, Ken,
let's
it's
do
your
it.
lecture.
It's all a Chateliers principle, which is one of my favorites, which turns out matters for things like climate change.
It does, as a matter of fact.
Dr. Pinner, thank you for the chat. Thank you for joining us, and thank you for the resubscription. We don't need your support because we're just doing this for fun, but we do appreciate it because it helps our drinking habit. We get to try things like gin and tonic out of a can. So,
Thank you.
Dr. Pinner, thank you very much. It's part of our job, believe it or not.
Yeah, it is.
Kinda.
That's fabulous. So glad.
It goes in my NSF proposal.
Great. Yeah, I have an NSF report, and guess what? I'm saving to turn it in until I have the link for this.
I think I got you drunk a couple weeks back. Oh, that's one of our guests. So yes.
Oh, no. Oh, shit. Who's
It
that?
was a couple weeks back. That's Lance Cooley, but
Oh, that's you,
Oh,
not
man,
me. I'm
I
like,
didn't.
I have not been drunk in the last couple of weeks.
oh yeah, that's a good question who this guest is, but or who this chatter is. But oh man, Lance, I think it came out to like 4 shots of whiskey that night, which is I was hungover the next day, but it was a good time.
Where was that? Was that here doing
Yeah,
science?
yeah.
Oh man. Okay.
Yeah, no, I mean, sometimes we hit it hard, sometimes it's casual. I mean, you have to wake up early tomorrow, so.
I really do. Yeah, I've got to take a teenager to the bus at 6:12 tomorrow. So I told Ken, this is not.
Yeah.
We're
No,
not
we're
gonna have
stopping at
that much
10:30
fun.
tonight, so I apologize.
Everybody was looking for. Yeah, right.
But yeah, that's so dumb.
Yes, correct.
So I
High
teach.
school is dumb.
Well, I teach Gen Chem 2 at 8 a.m. Monday, Wednesday, Friday when I teach it. And it's like every ounce of educational research says if they're under the age of 24, they shouldn't have class before 10 o'clock. But who needs research when you're teaching a science class, right?
When you're in the academy. Yeah. What are we doing anyways?
Yeah, exactly. Like, what's the point? But that's, man, high school, that's so brutal.
Yep. And I think Leon County voted a couple years ago whether to change the schedule to make it more, congruent with research. Mm-hmm. But, we all voted no.
I mean, but it's hard, right? Because work.
Yeah. So I think a lot, there's that. And then I think a lot of kids have after school jobs and then also for the sports, right? And so I think that was part of why so high school starts at 7:30 and ends at like 1:50. And then they have a bunch of after school sports and activities and stuff like that. That would be really hard to get that and all their homework done.
Yeah.
If they started later, which maybe just means we're having them do too much.
That's entirely possible.
I don't know.
Got to fill out that CV as a teenager.
Right. Yeah. All the nonsense.
Oh.
Yeah. Anyways.
I mean, that's crazy. Like your discussion of impending college is probably happening, right?
Yeah. So we have friends and neighbors who have seniors right now and juniors. And so, yes. And then my 14-year-old is talking with them. So yeah, all I can say is get a 529 plan. You have 529 plans for your kids, right? I'm serious.
No
I'm going to fund it right now. I'm going to give you $50 and make you start a 529 plan for your kids.
I don't even know what that is.
Oh my God. Okay. So we're going to stop talking about science. We're going to start talking about how you fund your
financing.
Kids. Yes.
I have not had an economist on stream. If anyone knows an economist, we need somebody. But
no, I'm telling you, this is the best thing. You got to sign up for these. So all states offer them. It doesn't matter what state you live in. You can sign up for any other state's 529 plan. And you... how does it work? You send in money and it's a savings account for your kids to go to college. And you pay taxes now on all that money, right? But then when it earns interest tax free. And when you take it out, if you're just paying for college, it's tax free. So, it's only taxed right now, on as part of your income, but then it's not taxed again later. And so, you need to start these right now for your children.
So that's a per state. They have to do in state, or how does that work?
No. And so we went with the Utah one, and it had good,
Or you just searched for the best rate and that was it?
Not even just rate, but sort of reliability and I don't know, different stuff. So Utah was highly rated, but they absolutely do not have to go to college in Utah. And it, goes wherever. So you want to fund that right now. You want to, if you have parents who send your kids a bunch of crap presents, what you want to do is ask them to not put money at that garbage, but instead put $25 instead towards the 529 plan.
Yeah.
And that's what you want to open tomorrow, and I'm not kidding.
So what happens if they don't go to college? Cash it out at a penalty?
You just pay taxes on it.
Okay.
Yeah. So
you better go to college.
Yeah, exactly. So for the love of Pete, everybody who's out there, if you have children, start this now.
Okay.
Yeah.
I'm going to completely forget all that, but we have a YouTube video.
I will email you.
Actually, we'll cross Debbie's path on the way out. If you remember, tell her this, and she will be excited.
This is what you need to know. Start a Utah 529 plan.
I'm going to be completely honest. I don't handle any of the finances in the house. I don't even know how much money I make.
That's for the best.
Yeah, exactly.
That's fine.
I'll just play video games.
Yeah. She'll let you know when it's a need to know basis.
Exactly.
Yeah, that's smart. Yeah, it ends up being a lot of divide and conquer, doesn't it? Like you just have to survive with the kids.
Pick and choose your battles and your expertise.
God. Yeah. All right.
It's actually tax deferred when you put money in. So Scuzzbot 10 is an actual tax expert. So
okay, well, I'm glad. I'm trying to think. I don't know that we're getting. Well, at any rate.
Ooh, Scuzzbot, what's your opinion? Should we do it?
Oh my God, yes, right?
Yeah.
I think Obama called this like the biggest scam or something. Like he kind of wanted to shut it down because it was so much. I think he called it like free money for college, and he thought it was kind of unfair. So it's like it's that good.
Was that like oh no, don't do it because it's unfair, or was it?
Yeah, just like basically, if you can afford to do it, it's like free money for your kids to go to college.
I mean, that's hard because then it's like a rich get richer scenario, right?
It really kind of is, but like, if you want to talk about what college tuition is like, having had these conversations with several friends in the past week, I'm like, I'm gonna go check my balance on their 5 kids 529 plans. So you want to do this?
Yeah. Well, the other one, college enrollment. So I was at University of Minnesota last week, actually visiting, and I visited a couple PUIs there. But like most of the Midwest schools, their enrollment is down 10 to 40%.
Well, there's this whole that's amazing.
That's crazy.
But Chronicle of Higher Education has been talking about this, right? There's a coming demographic shift, and I think the pandemic accelerated it, right?
Yep.
And I think places like FSU will be okay, right? These flagship schools, but there are a lot of institutions that are not. And I mean, it's this existential crisis in academia, right? Like we take it from the left for not being inclusive, and we take it from the right for not being relevant. And I think we have to address at least one, if not both sides, for self-preservation, right?
Well, and I mean, one of the fundamental questions people are asking right now is how much is a college education worth? And should you take $50,000 a year in debt to get the same degree you get at FSU? No.
Right. Well, this is the conversation I've had with some friends and colleagues in the past week. They're like, we're consulting with financial advisors right now to try and figure this out. And is it really worth it to take out X amount in debt so that the kids can do this? And is it worth it? And man, it's bonkers, right? Like these are not simple choices. And so open your 529 plan. It's going to be my PSA all night long.
It's brought to you by 529 plans.
That's right. I'm like a walking advertisement for the Utah 529.
No, I respect that. If you stand behind something, by all means. I've not even dove down that rabbit hole.
Yeah, well.
There's only 6. We're fine.
No, you can. Where's the financial advisor on here? Come on, back me up. Come on, help me.
That's your niece.
Get on
Fix
this. Yeah.
I'm not expected to do everything.
Seriously, it's exhausting. Oh my God, Ken. I am so tired. I can't even.
All the time.
Jesus, where's my gin and tonic?
Man, exactly.
Christ.
Well, let's dive into that rabbit hole. Do you ever debate whether this job is worth it?
Oh my God, we can't talk about that. We'll just cry. I'm so tired right now. You can't do this to me.
On screen,
no, just give me a minute. So I was telling Ken before. Oh my God, you're totally gonna make me cry. I swear to God, anybody who's on this project will understand.
It's just everything all the time. Everything we do is the most important thing ever.
Jesus
every faculty meeting,
Christ. No, they're not. The faculty meetings are definitely not. No, so we were on this great boat for our cruise in. Oh my God, I can't Tetris talk at the same time at all.
We can change up games if you want. We got do Mario Kart, Super Mario World.
Yeah, we'll change it up in a second.
Okay.
But the boat that we're supposed to go out on in June, July has too deep of a draft to sample our most important nearshore stations. So we've been working super hard, friends, to try and figure out how to make this work. And boy, has it consumed my past, 6 weeks, like completely. Like, oh my God. And there's no perfect answer. It's just really hard. So, you want to make me cry. I'm going to start talking about logistics. And I will start crying.
Well, the thing I wrestle with, and I mean, not those particular problems, but we spend a lot of time troubleshooting these things that are major issues in our endeavor. But like when we look at our publication record or the actual impact of our science, like it's probably going to be nominal. It might be huge, but it's probably not going to be huge.
Right.
Our legacy is most likely the students that we produce and their expertise.
Right.
But there is a cost benefit to like my tenure tour. I lost 10% of my 1.5-year-old's life to traveling.
Wow. Yeah.
And it's like,
our department doesn't pay for a tenure tour. We don't do that.
So we get teaching relief, but like chemistry's lucky because we like invite speakers. So I literally just emailed a bunch of people saying, "Can I come?" But it's time. It's an investment.
Well, and oceanography is like that, right? So I was on a 6-week cruise when my oldest was 18 months, and I just cried for the first 24 hours that I left her.
That was the first time away.
Well, it wasn't my first time away, because I knew enough to like not make that my first 6 weeks. The first, like that.
Start one day and then 3, and then
I didn't have enough time to like do multiple, but I did like a long weekend, visited a friend somewhere or something like that. But yeah, it's rough, and every day, and like, man, so my husband and I are both faculty, and the two-body thing is just so hard. And lately we've been talking about a lot, like the journey, and it has been long and hard to get 2 tenure track faculty positions and make it work. And it was a long, slow path. And then people like to ding me all the time for not having been productive enough when I didn't have a tenure track job or startup or facilities to do my work. And yeah, it takes a toll on your CV, and you're having babies, and so it's hard. Like it's a hard.
You'll have to forgive me. I didn't know your husband was tenure track.
So he's professor. Faculty. Yeah, he's full professor of physics. So he's theoretical astrophysics, does cosmic microwave background radiation. So he works with David Collins, actually.
Oh, okay.
So,
oh, that's fun.
Yeah.
I think I met him briefly when I visited your house, but.
Yeah, and then when we did the Ask a Scientist in Railroad Square.
Yeah.
So he was running around with the kiddos.
No, that's fun.
So yeah, but he makes it possible so I can go to sea. But boy, it's been a long, hard slog to get to. All we do is science and kids.
Don't worry, there's only like 20 years left of this shit, right?
Which part? The kids or retirement? Yeah.
Retirement.
Kids are out faster than that. Yeah, so we have a 9 and 14-year-old. So yeah, it's going fast. The 14-year-old is like, she's gonna be out of the house before we know it. And then 9-year-old, we have a little bit of time, but man, she's in this real sweet phase, and we're gonna hit teenager dumb before we know it, and it won't be quite as sweet. But right now, she's all sunshine and rainbows. Yeah, which is great.
I'll be content in this 3 and 6-year-old world for a while.
Yeah,
just like potty trained.
Yeah,
no diapers anymore.
There you go.
Like, still happy life exists. Still want to communicate.
Yeah,
but
enjoy it. And then like they're super sweet until like tweens, and then I think we're turning the corner with the 14-year-old. She's almost 15, so and she really is all sweetness and love. But
no, it's hard. They want independence but don't have it. Like, they recognize it.
Yeah.
But aren't functional enough to exist independently.
Here's some of that.
I get it. And we've had developmental psychologists.
They're on staff.
They're one phone call away. Like, sure, I can lecture on equilibrium and CO2 cycles, but they like study humans.
Turns out that's not useful when you're trying to parent. Yeah. Turns out we know nothing. Parenting's hard.
I mean, that's the hard part, is all the research is statistical distribution, and your kid lies somewhere on that
and they're not part of that. They're
4 sigmas off.
Sigma out. Yeah. We're like, nope, keep going, not ours. No, keep moving.
I mean, the reality is that they do fall in that distribution, but it's like the weird little things. Sometimes, yeah. Those events when they're outliers. The weird ones.
Oh my God. We should go back to science.
This is science. We're just not good at it.
Yeah, that's right. Yeah, that would have been useful to study.
Yeah. You think modeling the nitrogen cycle is hard? Humans.
Yeah, parenting hard. Yeah, nitrogen, whatever. I can figure that out.
Feed your kid nitrogen.
You do need to be, so we're vegetarians. We do need to make sure they get enough protein, but done. Easy. Like, that's fine. Had edamame at dinner. It was totally cool.
That's fun.
Get your kids a 529 plan.
So we detoured a bit. Let's head back to the science and expertise. And then
Ken's pouring another drink.
Yeah, exactly. I mean, this is my excuse to drink, really.
God.
And I don't teach tomorrow, so I'm fine. Yeah. But so one of the things I like to ask is if you had to like clarify something about your discipline to everyone on Earth, like you had a megaphone and you said, you know, let's, clear up this misconception, or like, I wish everyone understood this.
Okay.
What
easy.
is that?
Yeah. So when we call it a research cruise, it's not glamorous. It's not a princess cruise. We might shower. We're working 24 hours a day.
Why do you guys call it a cruise? It's so misleading, right?
Because what else are you going to call it? Yeah. So this has been funny. So you know how you can apply for funding through FSU? Yeah. I've actually had people be like, why are you calling it a research cruise? I'm like, because that's what the discipline calls it. They're like, that's dumb. And I'm like, you're so patronizing. It's annoying. But
not wrong.
I'm like, this is actually what we call it.
Did you go to the buffet? Did you see the comedy show?
Yeah. Oh my God. So some of the boats are very stinky, some, and then you're getting seasick and you're working 24. And like, I've been on cruises that are 6 weeks long. You don't get a day off. There's no days off. There's no overtime. You're not getting paid extra. Mm-hmm. So, it's not like a princess cruise. So that's what I have to tell people all the time is that research cruise. So now I think we're trying to call them like research expeditions or something like that, which sounds like we're going over land in Antarctica or something like that. But it's like, it's not it's not luxury, friends. Like 2 of us in a very small room, bunks, and small showers. And it's not.
So how's food handled? Do you have like a chef on board or what is that?
Yes. And we got so lucky on this most recent cruise that we were out on. It was, we had the best cooks ever. And like, we had 17 science party and 13 or 14 crew, something like that. And they were just amazing. We had people with all kinds of different allergies or sensitivities or, I'm vegetarian, whatever. And they just accommodated everybody. And the food was amazing. It was just amazing. Awesome. And it's not often like that.
All right. What game do you want to go with?
Let's do Mario Kart.
Mario Kart 64.
Yeah.
All right, so you are back to the crippler controller.
Oh, God. Okay, people, enjoy. It's going to be ugly.
Well, come on. Maybe? See if this hawk wants to open. There we go. All right, now we're on to Mario Kart 64. Sylveon, go Knowles. Yes, FSU represent. Welcome to the stream. Welcome to the chat. Thank you. First time chat. If you have questions for Dr. Angela Knapp, she's happy to answer them. Anything oceanography related, as well as
financial advising for your children's college.
529s. Open them.
I'm here for you.
Oh man, we've journeyed a lot of. That's what I love about this stream. It's like it starts out like really technical science questions, and then 3 drinks in, it just goes wherever the hell we want it to. So, Sylveon, welcome to the stream. If you have any questions, throw them in chat. We are happy to address those, or at least try to answer them for the most part.
My 9-year-old insists that we only pretty much do Moo Moo Farm, and that is the only track that we run. So anytime I have an opportunity to do something different, it's good.
So I don't know if it's the drink or I was troubleshooting the video game, but what was your one clarifying thing you wanted for everyone?
It can be so ugly. Oh, it's not a research. It's a cruise. It's not a princess cruise.
Okay,
It is work all the time, and it stinks, and we don't shower.
I mean, has your community discussed this nomenclature? Because cruise has a very popular connotation.
No, because we're all too busy trying to science, man.
Fun fact, though, from Reddit thread recently, I learned every Disney cruise or any those types of cruises, they have to have a morgue on board.
Jesus.
Yeah,
It's
so
the
somebody-
freezer.
Yeah,
Right? It
yeah.
just doubles as like where
Well-
your meat is. That's all it is.
I think they technically have to have a separate
No,
one
I
for
don't think
humans.
so.
But
You
like,
have no idea the space constraints these boats are under.
there's enough old people that they just
Oh, Jesus.
like it happens. Like every single cruise, they lose one person, and they have to debate whether they want to, pay for them, ship them back
God,
from
to
a
go
port,
in. Yeah, right.
or keep them on board. And
Yeah, these are like the science questions we do all the time.
yeah, exactly. Yeah. So enjoy your cruise.
Yeah, right. We have a dead freezer, but it's for preserved phytoplankton that are preserved with like formaldehyde or whatever. So that's our version of a dead. It's in the labs. I also, well, maybe I don't know if I'm allowed to talk about this, but so I don't know that this is true, but I have a theory that on the first cruise I was on, that there was somebody in witness relocation program, which I think is like actually the best place to put somebody.
On a ship.
Right. So it's a federally funded thing, right?
Yeah.
These are, the boats are technically owned by the Navy, but they're operated by the National Science Foundation, and they're usually affiliated with the university. But, and he was the cook. And I swear he was witness relocation. He said his name was Vinny. He was from New Jersey, and he lived a bad
Horrible
life.
accent.
Yeah. Oh my God. And he called me Angelina or something. He wanted to believe he was Italian. He wanted to believe that I was Italian. I'm not.
He was a bad cook.
I don't remember. It was so long ago. I don't remember.
It's like Red October, the cook.
Yeah, right. Exactly. No, he was very sweet, and he's like, no, in my youth, I got into a lot of trouble. I did bad things, but I've straightened up. I've changed my ways. I'm a good guy now. And I'm like, oh my God, you're totally witness relocation. Which I think is just
Bad.
the best place. They're at sea, right? Like, and it's close. Like, we don't, there's no ship to ship transfer unless something really exciting is going on. And I have had that happen too. But if there's a medical emergency, you can have that. But like, he would be totally protected out there, right? And like, it's scientists who are out there. Like, they don't care about any
Cuddle Puppy wants to know, did he look like Ray Liotta?
No, he was older and shorter. Well, I guess Ray Liotta's old now, but yeah. So, yeah. And his name was Vinny, right? So, like, totally,
That's fun.
totally witness relocation program.
Cuddle Puppy, you got a lightning bolt in 4th place, confirmed hacker.
What? I want to repeat, this is not my normal controller. Usually I'm very good.
You're 1st place right now.
Oh, I am? Okay, cool. So I don't... Advanced.
Cuddle Puppy, welcome back to the stream. It's great to have you. If you have questions for Angela, she's happy to answer them. Marine nitrogen cycle, oceanography, sampling phytoplankton at sea.
Don't we have some more questions for people?
Yeah, we'll take them as they come in. Whoever has questions.
No,
throw
I
them
mean,
in
like,
chat.
you made me all those yellow, the things I sent you.
Oh yeah, the predictions.
Yeah, the
Stupid.
predictions. There
I
we go.
forget them. All right, ladies and gentlemen, we're gonna put up a prediction. I like the first one. I'm gonna
Okay.
do that one.
Oh, yeah, that's a great one.
Yeah, that's a really something not intuitive and you can think about it but not have any idea of scale. All right, ladies and gentlemen, if you're not following us, click the follow button. 1, it helps the visibility of our channel, but 2, it gives you standard internet units that you can gamble on predictions. So we're going to do a science question right now. We're going to throw that up. The question is, what % of atmospheric oxygen comes from phytoplankton? Basically, the biological creatures in the ocean that make oxygen as a byproduct of their photosynthetic processes. How much of that % of atmospheric oxygen comes from the ocean versus land biological species? So is it 20% or is it 50%? Throw your prediction in there now. You are on Ask Scientist Gaming Honor Code, so you can't search the answer. You can't ask Chat GPT. Just make your best guess based on, I don't know, intuition, what you know about marine oxygen
How hard
cycles.
the phytoplankton are working. Who loves a good, hard-working phytoplankton? Raise your hand.
Are they in bloom or are they not? We don't know the answer.
Come on.
But yeah, and Dr. Pinner redeemed Take a Drink. So cheers, Dr. Pinner. Oh, Angie's in 1st place.
I'm
you can't do
working.
this. Is
I
that
gotta,
intentional? Because she's in competitive mode. Right? Is this last lap? It is.
Oh, is it?
Yep.
It's good. Somebody's paying attention.
Yep.
Oh, I'm ghost.
That does not help you at all.
No, I need... Oh, come on, Yoshi.
Oh,
Oh, don't crash into the wall. Go. Sorry. Okay.
Good game. Well done.
Me and Yoshi are buddies. What can I say?
But cheers.
Oh
You
yeah,
need
sorry.
a drink.
Here, I've got a little bit left.
Do you want to try the can?
No, I'm not gonna have a whole can.
Try a little bit of the can.
I'll have just
I've
a.
used her.
Oh, okay. All right. I'll have a smidge of the can. Yeah.
You'll have to let me know. You're the connoisseur of gin. So I'm always curious because there are quality control chemists that are working hard to make this drink taste like gin and tonic. So let's do a critique.
Seriously,
of how
yeah,
well
we
they
can
do.
send some feedback to Bombay and maybe. All right. Cheers. Thank you for the.
Dr. Pinner, thank you for joining us.
Yeah.
Thank you for getting us drunk once again.
Here we are. That's very different than yours.
Yep. I'm guessing it has a whole lot more of the tonic water than it does gin because it's only 7% alcohol. I feel bad. My guests have to go home after this.
Yeah, take teenagers to school very early in the morning.
That's so dumb.
Oh, well, I'm not even paying attention. Look at this.
So the thing I'm wrestling with, and I might send out a survey because I haven't scheduled the summer yet, is like, what night should I do Ask a Scientist Gaming? Because Friday and Saturday would be better, but I don't want to take people's Friday and Saturday,
right. Yeah. Don't know.
Cuddle Puppy. Better question. What % of methane comes from phytoplankton? Don't laugh at that.
So rude.
don't encourage Cuddle Puppy. Phytoplankton. There's your new NSF proposal.
Yeah, nope.
Hey, you can study farts on your cruise.
Yeah. So phytoplankton don't produce. Well, so here's how they do produce methane: is dissolved. I'm going to totally go. Yeah. Dissolved organic phosphorus is an important phosphorus source when phosphate is scarce, and their methylphosphonates in the surface ocean and phytoplankton have been shown to use that as a phosphorus source, and in the process they produce I'm going in there's actually a near surface methane peak, and so there are a lot of people who are working on this and trying to understand how important that is as a source of methane. Oh God, come on, I had a star. Jesus. To the atmospheric methane burden. So that is how phytoplankton, they're not farting, but they are accessing a different form of phosphorus than they would prefer to use. Here we go. I need the lightning. Why didn't I get my lightning? What happened to my lightning?
I think you did it, didn't it?
I just crashed. Yeah, but then it made me crash.
It made you crash.
That's right, Ken. It made me crash. That's correct.
I'm not a computer scientist, so I can't.
Don't worry about it. Come on now.
Cuddle Puppy. Scientists prefer to spend their Fridays and Saturdays getting blackout drunk and starting bar brawls. Oh man, when I was a postdoc,
I am not hanging out with the right people.
when I was a postdoc, we had it was every Friday night, just a gathering of like a whole lot of grad students and postdocs drinking. UNC Chapel Hill back bar. It was pretty spectacular. Foosball across the board.
I'm going in the drink again. So at Princeton, there's a graduate student dorm. First of all, you need to know that, and that is where my husband and I met. The male to female ratio in that dorm, it took me a while to figure out. By the end of the first week, I did. It's 75% male, 25% female. And the saying is the odds are good, but the goods are odd. Yep.
Georgia Tech has the same thing.
I see. But there's a bar in the basement called the D Bar, the D Basement Bar. It was actually a really good bar, and it's very cheap. I think drinks were like at cost, basically, and they had a really good selection.
Wow.
Yeah.
It's like university subsidized drinking.
I guess so, because it's a private school, so they can do whatever they want. Yeah. And yeah, a lot of people would show up because it was, good. But that was back in the days when smoking was still legal, and so it's disgusting. Like you come home stinking of smoke. It was gross. Yeah. And then you go back to your dorm room. So did that for 1 year and then you move out.
All right, so we're almost at the 10 o'clock mark. Are you ready? So we're closing tonight at 10:30 because Angie has to get up early tomorrow because high schools start too early and it's stupid. But that is beyond the scope of our control. So we're going to close at 10:30, which means Angie's going to be starting NARC in...
Oh, God.
Are you ready for this?
Sure.
I mean, unless you want one more race, it's up to you.
Yeah, let me finish out
Just
Mario Kart.
Just do one more? All right.
My happy place.
Go ahead, because it's going to take you about 30 minutes to do NARC, plus or
Really?
minus.
Oh, okay.
I mean,
All
the
right.
world record is 9 minutes and 4 seconds. So,
He didn't tell me this before I got here tonight, so I was not
no, I didn't.
prepared.
I mean, it's hard because like you're a veteran guest, right? So I didn't have to ask for
Veteran, I've been here
bio. I
once.
didn't have
Yeah.
to ask for like we knew what we were gonna do, so I didn't give you the heads up. But yeah.
No.
Oh, Scuzzbot 10 has redeemed request a factoid.
Okay.
Whether it be financial or oceanography related, it's totally up to you.
Going in. Trichodesmium are the best. They're my favorite charismatic phytoplankton species.
What
They-
makes them charismatic?
Well, it's because they break all the rules and we don't understand how and why they do all their things that they do. So they fix both carbon and nitrogen. And of course, when you do photosynthesis, oxygen isn't this. I'm totally dying this. I should have gone to NARC. But oxygen is a waste product. But here's the thing: oxygen permanently denatures. It breaks the enzyme that carries out nitrogen fixation. And so when you have this going on in a bunch of single-celled organisms, the question is how and why. And some phytoplankton, basically the answer is temporal or
Take that, Bowser.
spatial, that's right, separation of the processes. Oh no. And Trichodesmium may or may not have temporal and spatial separation. We can't quite figure it out. They probably have some temporal separation. We don't know how they do it. They are amazing. If you know what you should do right now, Ken, is you should Google a video and it will break your brain. It is, if you go to YouTube and you Google Yela, Yeala, Shaked, shake with a D on the end.
Sorry,
And-
say that again.
Yeah,
What-
Yale.
why?
So it's this it's an Israeli colleague. She did
Yela.
Shaked, Shaked, and Trichodesmium and iron.
I don't
So
know.
trichodesmium, yeah, Trichodesmium, and iron. And there's a video that will break your brain for what these phytoplankton do when they're given dust. This time from
Oh, it autocorrect to Yella Shaked.
that's no,
what's the last name?
Shaked. So shake with a D on the end.
Shake. Okay.
Shaked, yeah.
Shaked, trichodesmium,
And
iron.
dust. Put dust in there. Whoa, I'm totally not making it off. I am dying this round. Sorry, guys. Should have gone to NARC.
Is that the video?
Yes. Oh my God. Show people this.
All right, let me
These
see if
are
I can
plants, people. They don't have brains.
see if I can get a browser
It's
open here.
Unreal what they do.
These are plants that have like mechanical motion.
These are filamentous cyanobacteria, and they are being given dust. And dust is important because it has a lot of iron in it. And so the idea is that these phytoplankton are taking dust particles and they're moving it to the interior of one of these colonies, which may be anoxic, and probably they're producing a lot of ligands and sterophores that can solubilize this otherwise super insoluble iron and turn it into something that they really, really need to carry out nitrogen fixation. But like, how do these not look smarter than your average human, if not faculty member? Like, they look so much smarter, and they just are, right? Like, it's bonkers what they're doing. They're just amazing. And so we found so many of these on our February-March cruise. We're so excited. We found all of them. We have all kinds. Our team is just super well equipped to try and figure out what they're doing and why they're doing it. And it was also interesting because they usually live in warm waters. They usually don't occur in waters less than 25 degrees C, but we were in waters that were 22.8 degrees C, and they were super happy. So I was telling my colleagues they need to write papers and describe our cold weather tricho species, and we're going to have them be our mascot, and we're going to draw little pictures of them with a hat and mittens on and a scarf, and it's going to be adorable. trichodesmium. So much. It's all the much.
So, building upon that, the charismatic trichodesmium.
Yes.
These are the phytoplankton that have slicked back hair and leather jackets.
That's right. I guess. Yeah, whatever does it for you.
I generate an image of that, I promise you.
Oh my God.
Huggy Beer, welcome back to the stream. So this is what turns us into zombies? I mean...
They do release neurotoxins. Does that do?
Huggy Beer, you were right. You predicted World War Z.
Yeah.
Here it is.
Yeah. So.
It's pretty crazy.
Yeah, right? How are those
plant life that tracks anything is amazing.
Isn't that amazing? And it's like pulling it into the middle where we think, they're so smart. They're so much smarter than us. Okay, you can end Mario Kart. That was not.
All right.
I ended up in third.
That's not bad.
Not bad considering how many times I went in.
All right. Are you ready for this?
No, definitely not.
I mean, the thing is, it doesn't really matter, but it kind of does.
Pressure. Okay.
All right. So, ladies and gentlemen, we close every stream with NARC. We have our guests race against the clock. So, Angie, go ahead and press start.
Before. Okay. Oh. It tells me I just go forward and walk through doors.
Yeah, and you're going to win the war on drugs. So, this is totally worth it.
So glad. Okay.
All right.
Oh, jumping.
Yep.
Oh, there I am shooting. Okay.
So yeah, anyone not familiar, we close every stream at Ask a Scientist Gaming with NARC, a bargain bin game from the 90s that's 1980s, that's absolutely terrible. But I started playing it, so I make every single guest play it. Angie was kind enough to agree to play this game. No one says no to me, so...
He's giving us drinks.
I give him the heads up.
What am I supposed to do
Get
with
the
this?
card.
How am I supposed to
pick that up.
There we go. Okay.
Just keep going.
This is my kind of game, friends.
No, exactly. This
walking.
is you're gonna die.
Eaten by the dog.
So you have infinite lives.
This is tough.
You have infinite bullets.
Thank God.
Yeah, infinite rockets. You're fine. You just got to go. Like it's literally just keep walking right, and you'll be fine. But anyway. All right, Huggy Beer says, I'm not sure how off topic this is, but why is it so hard to get to the depths of the ocean? I know pressure, but with drones, should we be able to make drones get down there with a camera?
Drones. Yeah, I think drones can go. But I mean, we're not there for pictures. We're there for the science, man. So, so there's some things to learn by pictures. But,
I mean, yeah, I was going to say in their defense, like some sea life, pictures is enough to,
I guess.
file them
I'm here for the chemistry. So, right. Good question. So here's the deal. So we can definitely send equipment down to sample. It's like, keep going.
Keep going. Keep going right. The doors will be obvious-ish. There are doors, but there are doors that matter.
Not that one. Liquors? How about liquors?
You want a loan?
Keep going.
529?
Yeah, exactly. Where's the
529
shop? Go there. Ken.
I choose to be a bad parent.
No. None of that.
All right, but they are making drones that go in the ocean.
Yeah. So we can definitely send equipment down deep enough. Usually, so I am often one of the
Sorry, I'm going to have to intervene on this.
Yeah.
So you have to shoot the guys wearing black with bullets, not rockets. So just holding the shoot button. And then one of them will drop a blue card.
Okay.
So
I have to keep
you have to hold
Yep.
Oh, God. This is one of the parts where you have to actually kill them.
Oh, jeez. Yeah, see, not part of the deal. Didn't tell me that.
No, I didn't. That was not part of the walk right tutorial.
Yeah, that's right.
There it is.
Oh,
All right,
thank
go
God.
through the door.
Okay. Yeah, let me leave. This door? No, apparently not. Isn't it? There we go. Thank God. Okay.
Laurie, welcome back to the stream. The drink this evening is gin and tonic. We are using Plymouth gin, and I don't remember the tonic water. I'm not going to look it up, but yeah, gin and tonic tonight with a slice of lime, an actual lime. We didn't use the liquid lime.
No.
So yeah,
high quality.
We keep it classy.
Yeah.
But anyway, drones in the ocean.
So we can totally sample the bottom of the ocean using the equipment that we collect water with throughout the rest of the water column. I'm usually the only one. Like if I'm on the boat, I'm telling people I'm sending our package, the CTD package that collects water, down to the bottom. It usually annoys people because it takes a lot of time, and not everybody cares about characterizing the water column the way I do, all the way down to the bottom. But if it's my cruise, guess what? We're gonna do it. But
Go back, see that car?
No.
You can drive it.
Oh,
So
great. Okay.
Tap B, jump in it,
Okay.
and stay at the bottom of the screen. It'll get you a certain distance, and then you're gonna crash.
Why can't I get
tap
in
B,
it?
Tap B. There we go.
There we go. Okay, thank
There
God.
you go. Then you run him over.
Like,
Totally
I
worth
am.
it. Now
Oh
you're done.
no, now? Jesus. Okay. So a lot of boats, say you're near like the Puerto Rico Trench, which goes down like 10,000 meters or something like that. This guy, annoying. Go away. Leave me alone.
Just keep going.
Okay, Ken's like,
Just keep going. There's
Don't do it.
no value.
Don't do it.
You can outrun him. You're fast.
Jump him.
Oh, that's your move.
Button mashing.
Yeah,
That's what that
perfect.
was. That was button mashing. No skill.
Jump.
Oh God. What?
It's garbage cans. Jump over.
Oh, Jesus. Okay. So most of the time they don't put enough line on the CTD package to go down to 10,000 meters. It's usually a 5,000 meter line. And so that's usually what we're limited by for the kind of work that I do. Other people, if you want to go send somebody down, then we're talking about like some of the submersibles, so like Alvin. So I have a colleague, Amy Bacco-Taylor, who goes down in submersibles a lot. She does deep sea corals, and her husband actually works on some of the and supports some of the technical staff that supports some of the submersibles. But I'm not usually a submersible kind of gal. I don't really need to go down. Oh Lord. Why are there dogs chasing me? I don't appreciate that.
So you can hold both buttons and shoot them. There you go. And they turn into little dogs, as physics dictate.
Right. That's how that works. Okay, good to know.
I mean, so they can go to like the depths of the Marinara Trench. Like that's
We could, yeah. So I don't think there's usually a problem because I've gotten samples from the bottom of the Puerto Rico Trench before. So I don't think that that's a particular challenge. It's a car.
So stay at the bottom again. You'll eventually run into something, but it's fine. Totally worth it.
Come on, let me in the car.
There
There we go.
Oh no! Valuable time. It's just costing you.
There we go.
Okay. Bottom for a while.
Nice. Okay, here we go.
All right, then you keep going. Unless you memorize this pattern, you're absolutely. So,
Which you have. But
I have. I've played this game a lot.
there we go.
Cuddle puppy, you want to tell her, or should I?
Oh my God. Nice.
How many Cthulhus have you studied in the ocean? That's the like deep sea creature, the kraken, godlike deep sea released.
None, but if I had one of my, is Kate Kuba out there? Kate, are you there? No, I haven't studied any of those. I am strictly a chemistry kind of gal, so I don't, or the phytoplankton, maybe some microbes if I get real excited, but no actual bigger life forms. No
I did say Marinara Trench, not Marinara. I apologize, Cuddle Puppy. Thank you. All right, Huggy Beer has a science question. How do you look? Stay on the bottom. You'll get all the way to the end. Perfect. What do you look for in the water column test? Like, what would be your eureka moment with that? That's a good question. You don't get a eureka moment on the boat, right?
You are right. I don't usually. So I bring my sample. Whoa, dancing! Look at that, kinky pinky. What the hell? What just happened?
It's wholesome.
I don't understand what's happening right now.
The war on drugs. Did you not pay attention in Dare?
No, we did not cover kinky pink. Oh my God, throwing shit at me. So my eureka moment probably wouldn't come on the boat, although we did get a little excited on this cruise. We weren't paying. Oh my God, look at this. Wow. All right.
So you're going to go in the top right one there.
Okay.
You're going to find a card.
So we crossed a low salinity feature, which, these guys.
So you're going to get that white card, and you're going to go through the door and press up, but don't press up more.
no.
You have to go to the door
Don't
again.
do that. Not like that.
It happens to everyone. No
You're
worries.
trying to save me. Oh, Lord.
So don't press up.
No. No. It's going to be like Groundhog's Day.
This is where we are.
infinite loop.
You thought you were going home tonight.
Yeah, I thought. Okay, barely up. Woo.
All right.
How do I get out
Go
now?
left.
Okay.
Yeah, sorry, you're going left. There's a key door right there. See that?
Oh.
Card in there, through the door. You're good.
It's a lot of pressure. Yeah, so I bring my samples home and analyze them, so I don't know if I'm having eureka moment at sea usually, although honestly we found so many trichodesmium, I knew I was pretty excited on this cruise. We just got, oh God, I'm talking science.
Was it worth it?
I don't know.
Right.
You're going to tell me.
I'll take that.
Oh Lord. And then,
Through that door. Now you're going to go left.
okay.
So you're going to shoot these green guys with bullets instead of rockets, and one will drop a green card. So hold the shoot button. I don't know why it requires bullets instead of rockets, but it does.
There's a green card. Here we go. So,
oh, there you go. Nice. All right. You've had some good RNG. The random card drops in your favor.
That's what I'm here for. Oh, here we go.
It's the only reason.
So we weren't quite paying enough attention. We were very busy. But then we realized that we crossed a low salinity feature, and we're not sure yet. We haven't had time to go back and figure it out if it was the Mississippi River, which usually flows to the west this time of year, but sometimes breaks right and flows around towards the West Florida shelf. Or if maybe it was the Apalachicola, we don't know. These doors? Not these doors.
Nope, keep going.
Okay, keep going. So when we crossed the low salinity feature, we were excited because we definitely wanted to sample Mississippi River water, whatever riverine water. Oh, jump. Fight. Was coming that we crossed. And so then it was actually kind of funny. We drove the boat back and forth over trying to find the low salinity feature for a long time. And there aren't satellites right now that are up that measure sea surface salinity, so that sucks. Would have made our life a lot easier. But there are sea surface temperature and sea surface height and other things. So we were trying to look at remote sensing products in real time to figure out where we should go. And we spent a lot of time driving. We spent a good chunk of a morning driving around. Oh, I need to kill somebody to get a card.
Nope, you can get that. You have that card.
Sweet, but bad guy's going.
So, Angie, I hate to break this to you, but you will not break the world record.
You're kidding.
Nope, it was 9 minutes and you're at 11.
Broken. Hit me right here. Yeah.
Devastated.
My ego is crushed. They won't promote me to full professor.
Yeah, no,
it's going on my permanent record. Yeah. I will not live this down. Yeah. This is sabotaged, Ken.
So you have this card.
Oh, do I?
Yep.
The dog, though. Oh my god.
All right. Go right, find the door.
Fast as I can. If I hadn't gone up so many times, I would have gotten out. I would have gotten a record. Oh my god.
Oh, that's fun.
Oh,
Huggy
no,
Bear
I'm
wants to
dead.
know, does the Mississippi River actually change the height of the Gulf after it hits open water? I mean, it's continuous flow, isn't it?
It is. So what does change the sea surface height is the loop current and eddies that spin off. And so that's where we see those sea surface height ends up being diagnostic for eddies and things that spin off. Yeah,
Just go
going
right.
the wrong way.
Just go right.
Dogs.
You shoot the dogs. Hold both buttons. Hold both buttons. There you go.
They're just destroying. I'm just
They're
a
relentless.
bloody pulp. It's awful.
What are you talking about? This game's amazing.
It is the best. Yeah, I take it all back.
I was going to say, you have this on an emulator at home.
Yeah.
so
Oh,
you
do
can-
I? There might be a reason the
All right, so now you're going to shoot the guy in the wheelchair.
Oh,
With
right,
a rocket.
Of course.
You know,
Yeah,
that's
like
the war
you
on
do.
drugs.
Yeah. Okay.
Did you see the movie?
No.
I don't know.
Oh my
The
God.
tapping A is rocket. Yeah, there you go. And you just got to time it up so you hit him.
Yeah, it's not gonna happen.
It'll happen eventually. You don't
Long
get to
enough, we
home.
got enough time. It's tomorrow. Sorry, kids.
Yeah, you didn't need that day of school anyway.
Yeah, that's right. Don't worry about it. Yeah, it's a
I mean, so when a hurricane hits, how much does that screw up your day? Like, just like a reset button?
We refuse to go out. We don't even request cruises in the Gulf in August or September, or
Just 'cause of the
preferably
hurricane
October.
season.
Like, heck no. Because you can't even plan. Because
Yeah.
the boats can't go out. And you don't know where it's going to go
there is, that's
exactly.
one.
Okay.
So you need
there's
3.
more? Are
Yeah,
you kidding
he's
me?
got an
This is
inchworm.
Garbage. Okay.
Game's so unrealistic.
Love it. Yeah, so no, we just refuse to go out. We will not do that. And I don't think so the organization that schedules the oceanographic research boats is the university. What is it? University
Ooh,
Naval
nice.
Organization.
That's 2.
Something. Oh, great. I don't even know what UNOL stands for, but at any rate, they probably would not let us. None of the boats would sign up to take us out in the Gulf in August or September. I think everybody knows about it. So, yeah, it's.
I mean, so in
Not
broader
smart.
picture, just like mapping hurricanes versus blooms, like is there a relationship? Does that reset the cycle, or is
Such a good question. Yeah, so I think that there has
nice.
been
All right. He'll
some
drop a gold card.
super doggies stop, leave me alone. Dogs. I think there has been some evidence that hurricanes actually basically entrain nutrients from the subsurface into surface
That's
water.
interesting. It stirs it up.
Yeah, exactly. And so then there's a phytoplankton bloom often that follows.
All right, I'm going to intervene on this boss. You're going to want to go to the top of the screen. You're
Yeah.
going to jump, and you're going to try to shoot him in the hat.
Oh
So you
my
have
God.
to jump and rocket him.
Oh, Lord. Ken.
Oh, there it is. Okay, rocket him in the face. I can't believe you just got that on the second try. Speedrun strats, ladies and gentlemen. Angie was not messing around. She's played this for months. All right, so you get away from him, and you don't have to jump anymore.
He's chasing
Just
me.
turn
I
around and
can't.
shoot him in the face
Oh
with
my
a rocket.
God, I can't. Ken,
You just
ran
got
me
through
over.
the hard part. Like usually I have to do that for guests, and you got it on second try. So congratulations.
My
If you
mad
achieve-
skill set.
If
Yeah,
you
what can I
achieve
say?
nothing else, you've done
If I
that.
quit, I'm done as an oceanographer. What do I have to do? Oh,
There it
look
is.
it.
Okay, so now you're going to run away from him and be like in the middle of the screen and shoot back and shoot bullets at him.
Oh, what?
So
oh my
go
God.
right,
I'm trying.
get a good distance, turn around, shoot bullets, move up and down, hold the bullet button, hold the A button. Yep, just like that. And you're just going to shoot vertebrae until they all disappear.
Oh my God.
And that is the remainder of the game.
Oh, he's just run me over. Oh, that needs
So
some
Angie's
distance.
been on the stream before, but we didn't start this tradition until after, so surprise.
That's the best part.
Exactly. Well, that's the thing. Like next time, you're gonna be ready.
Oh my God. Added degrees of difficulty.
So the thing we didn't say is, so this started out as a joke. I played this when I was the guest. I was like, this is a bargain bin game from my childhood. And I got a challenge to speedrun this game, which is beat the game as fast as you can. And I was the world record holder for about a year in this.
Yeah, you're actually really good at video games, right? Like you hold some records in like a lot of things.
I no, not records. This is the only one that I was like, literally, I could say I was the best in the world at for a period of time.
That's
And
amazing.
it's because I figured out the glitch on the previous boss to save 20 seconds through scientific method. But
Scientific.
I literally just played around with it till I found out what worked. So yeah, turn back, go the other, keep going right. Like you have a lot of ways to like make room.
Except now I'm smashed.
So yeah, so get away from him, shoot back, get away from him, shoot back. It was stupid as hell, and it's a terrible game, and I apologize, but do I? All right, if you guys have any last minute questions, we have about 10 minutes left while Angie tries to beat the second phase of Mr. Big. If you have questions about oceanography, the marine nitrogen cycle, phytoplankton, photosynthesis within the ocean, the oxygen generation byproducts, she is happy to answer any of those questions you may have. This is your last 10 minutes to do it before you win the war on drugs.
Oh my God.
I don't know if it's going to take 10 minutes. We'll see how it goes. Okay, so turn back.
Yeah.
So bullets are the only thing that can kill the vertebrae. Why do I know this?
World champion.
Studied that a lot in this game.
You basically have a PhD in this game.
Yeah, exactly.
You did a
If they
postdoc.
did grant a PhD. No, there's a YouTube video that has a tutorial that I made. It is the most in-depth YouTube video I've ever made.
That is hilarious.
My favorite part is I do a bunch of lectures that I put on our YouTube channel for a research group, and they're like on transient absorption and fluorescent spectroscopy and all sorts of stuff. And like my transient absorption video has like 10,000 views, which is pretty remarkable for a science presentation.
Yeah.
But my world record NARC run has 9,000 views.
It's fantastic.
So my expertise be damned.
You have dual expertise.
Yeah, exactly.
This one is recognized by the Academy, and the other is maybe not.
Exactly. All right. Oh, Huggy Bear has redeemed request a factoid. So you have a knowledge bomb while you're fighting the vertebrae. Turn back and shoot.
Factoid.
Hold A. Just hold the bullets. Up. Go. Keep going.
Oh my God.
Go right.
Factoid. I'm trying to think.
Under pressure.
Yeah.
No, you're not playing anymore. You just got to find a door, and then you have another level.
Oh God, are you kidding me?
No, you don't have to do anything on the next level.
Okay. Jesus. I'm gonna have sore thumbs tomorrow.
You earned it. Go right.
Yeah, I'm trying to get. I don't need the gold bars.
No.
Go.
Speed.
The world champion is sitting right here. Telling me what to do. Just listen to him.
Former world champion.
Man. So when were you the actual champion?
I was like for a year.
How long ago?
Like last year, actually. Yeah, no, it's true.
You have small children.
Right.
What the hell are you doing?
I'm committed to a goal.
Oh my God. Love that. All right.
Wow, so that's your first time playing at 1921.
Yeah.
That puts you at number 6.
Out of how many?
At least 50. Yeah, no, you crushed it.
That's pretty good.
No, your second try hitting the hat thing was no joke.
Oh, you guys.
That was
making me feel good about myself. Okay.
Sorry, there was a request to factoid. Do you have a
Nitrogen is the best.
that is an opinion and not a fact.
You are wrong. That is not true.
I'm partial to hydrogen.
Why?
It's
Justify your answer.
pivotal to everything.
As if nitrogen exists. Oh my God, we do this all.
The ocean exists without nitrogen.
Well, that's true. Okay, I will grant you that. Okay, so here's my factoid. We think nitrogen fixation was one of the first metabolisms on the planet. It had to be, right? To get all the nitrogen for all the enzymes that fuel life. So,
that's interesting. It had to be.
And we also think that because the enzymes that carry out both carbon and nitrogen fixation are actually adapted to early life conditions, so anoxia, when iron concentrations were much, much higher. So it's a really highly conserved enzyme. So nitrogen fixation was probably one of the very first metabolisms on the planet.
Man, that's
There's
pretty
your
fun.
factoid.
Yeah. For those of you that don't appreciate it, I teach this in Gen Chem too. It's like a major breakthrough in human development was the Haber-Bosch process, which turns N2 into ammonia, essentially. You take N2 and H2 and put it over a catalyst, you make ammonia. What's crazy is like half to 3/4 of the nitrogen in your body are direct byproduct of that reaction. We put it in fertilizer, it goes in the ground, and then eventually it ends up in your body. So nitrogen fixation is a non-trivial process. And yeah.
And your nitrogen isotopic composition is probably, you're carnivorous, yeah?
Yeah.
So you would have a higher delta N15 than me. So what we say is you are what you eat plus 3 per mil in nitrogen isotope studies.
So you could figure out degree of vegetarianism based on nitrogen isotope ratios.
Right. Yep.
Has somebody done that?
Yeah, so there's a bunch of food web studies to figure out trophic position in the food web. And so people use nitrogen isotopes of amino acids often because there's source and then trophic position, isotopic compositions of different amino acids. Yeah.
Man, that's really fun.
So I have a lower isotopic composition than Kevin. Sorry, but also, but.
No, that's fair. All right. Well, I mean, we're done 5 minutes early because you beat this game too fast. Does anyone have any questions, or is there any knowledge bombs you want to drop on the audience?
Nitrogen
so here's one we like to ask. And if anyone has suggestions on who we should raid, throw those in chat. We'll figure it out. We bring our people to other people. Like typically it's just like a shared viewership kind of thing.
Okay.
And it helps with visibility and whatnot. But if you had to like, well, what's the I don't know, flat earth or, you know, anti-vax equivalent of your field? What is the common misconception that people try to sell?
Right. So especially around Florida, when we're trying to understand what maybe causes the harmful algal blooms around the coast, there are a lot of people who want to pin the blame on the septic tanks or things like that, and they want to use nitrogen isotopes to do that because they want nitrogen isotopes to be a source tracer, but it's never just source. It's always source plus process. So it's always much more complicated than you want it to be. And you really have to have a holistic study of the environment to really to be able to interpret the nitrogen isotope signatures. So only use nitrogen isotopes responsibly and with an adult in the room, somebody who's highly trained, because you can't just pick a delta N15 value and think you know what it means. It's always more complicated.
I mean, so piggybacking on that, like, do you do political warfare within Florida? Because there's battles on how we should handle this. Like, is there, I mean...
No.
No,
I don't want to.
Well, you don't want to, but I mean, does science have a horse in this race? Is there a clear position that should be taken? Is there?
Well, I mean, I think in general, like septic tanks are probably not a great choice. You know, I think that's fair. But trying to understand where the fate of all that nitrogen is, I don't think that that's an easy answer. I mean, a lot of people want to say like Wakulla Springs is dirty because of the septic tanks, and it may very well be. But I wouldn't want to just use nitrogen isotopes, and you really need sort of a pretty detailed study to be able to understand all the context that goes into it and do the whole budget, right? Like you were saying, the elemental budget, like it's not just one time, one place, I'm measuring one species. You've got to understand what all the quantitatively relevant species are and the transformations that are impacting it and the inputs and outputs, and you get it.
Yeah. Oh, that's fair.
So yeah. So, and that's a hard thing to do, and it's expensive. And so a lot of people will be like, oh, well, here, I can tell you what the answer is for this much money. But I'm always like, it actually costs a lot more to do that.
It's hard to change your mind when your financing depends on it. But yeah. Yeah.
So I'm like, well, if you like, yeah. So if I'm talking to people, I'm like, I'm not going to give you the easy answer, but it'll be expensive and hard. So I understand if you don't want to work with me. Like, that's okay. I don't mind. I've got plenty of science going on. So
that's fair. Yeah. All right. On that note, we'll call it quits, everyone. Thank you for joining us at Ask a Scientist Gaming. Angela, thank you for joining me for a second time, this time in person. I really appreciate it. It's a lot more fun in person.
Way better.
Way easier. It's way more casual. Yeah. So thank you again, even though you have to wake up early tomorrow and you just recently got back from a cruise, a casual beach cruise.
You're fired. You're fired, Ken.
Oh man. I'll give beer. I'll address that later. Angela, any parting words for the audience?
Thanks for being here, guys.
Yeah.
Team Nitrogen forever.
Team Nitrogen Trichodesmium. Team Trichodesmium is here.
Yep.
So yes, in 2 weeks we'll have our next guest is Tanya Perez. She's an anthropologist that studies human behavior through archaeology and zooarchaeology. And so she looks at, the rich human history in terms of the archaeology that it's generated and trying to map like human behaviors based on that. And so, yeah, she'll be here in 2 weeks. That'll be May 10th. That'll be 8 to 11 p.m. So yeah, swing by for that. But it's always a pleasure, Angie. Thank you for joining me. It's been a great night. We've journeyed through many topics.
Thank you.
Does she have a bullwhip? I have no idea. Come back in May 10th and ask that question. But yeah, thank you for joining us. Stick around. We're going to raid somebody that does educational related things. But yeah, it's always a pleasure, and thank you for joining us.